ये कैमरा सेट करने के आपने। हेलो। 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 हेलो सर, वॉइस आ रही है हमारी? हेलो। 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 वॉइस आ रहा है? आप लोग बोलिए।
हेलो।
हाँ सर, गुड मॉर्निंग। गुड मॉर्निंग। वॉइस आ रही है सर? आप लोग बंद कर दीजिए। ठीक है वॉइस आ रही है। महेश सर। हेलो। हेलो महेश सर, वॉइस आ रही है हमारी आपके पास? हेलो सर, गुड मॉर्निंग। ये कैमरा कैमरा आप लोग को कैमरा इसको सही कहाँ है रिमोट?
वो जो पोस्टर दे दो, पोस्टर लगा दें।
हाँ पोस्टर यहाँ पे इसी को लगा देंगे सर। तो क्या कैमरा सही करना
हेलो दादाजी मी डॉक्टर भावना करता हूँ
ହଁ ହେଲା ଯେ ସେ କଣ ଏମିତି ହିଁ ଅଛି ତ No, unmute कर सॉरी म्यूट कर पण तोरू ये देखुଛି
हेलो एक मिनट मुझे ना
ಏನ್ ಮಾಡೋದು ನಾನು ಕೇಳಲಿಲ್ಲ ಅಂತ ಆಗೋದು. ಎಲ್ಲಾರಂದ್ರೆ ಯಾರು ಮಾಸ್ಟರ್ ಅವ್ರದ್ದೇನು ವೆಕೇಶನ್ ಅವ್ರದ್ದು ಸರಿ ಮಾಡಿ ಅಲ್ಲ ಎಲ್ಲ ಫಸ್ಟ್ ವಿಚಾರ ಮಾಡ್ಬೇಕು ಕೇಳಕ್ಕೆ ನಿಮ್ಮ ಹತ್ರ. ಅಲ್ಲ ನೀವು ವಿಚಾರ ಮಾಡಿ ಎಲ್ಲರೂ ಹೋಗೋಣ ಈಗ ಯೋಚನೆ ಮಾಡಿ ಅಲ್ಲಿ ಖಾಲಿ ಪುಕ್ಷಟಿಗೆ ಹಾಂ ಹಾಂ 2 days ಹೌದು ಅಲ್ಲ ಅದರ ಮುಂದಿನ ದಿವಸ ಈಗ 20 ಅಲ್ಲ ಸ್ಯಾಟರ್ಡೇ ಒಂದು ನಿಮಿಷ 23 ಹಾಂ 24 25ನ ಇದು ಇರ್ಬೇಕು ಅಂದ್ರೆ ಹಾಂ ಆ್ಯಕ್ಚುಲಿ ಸಂಡೇ ಹೋಗುದಾ ಅಂದ್ರೆ ಇಲ್ಲಿಂದ ಹಾಂ ಹಾಂ ಇಲ್ಲ ನಿಮ್ಗೆ ಇನ್ನೊಂದು ಹೇಳ್ತೀನಿ ಫ್ರೆಂಡ್ ಅಂತ ಹೇಳ್ರಿ ಇದ್ರಾಗಿಂದ ಮಂಗಳೂರವರು ಫ್ರೆಂಡ್ಸ್ ಬರತ್ತಾರ ಫ್ಯಾಮಿಲಿ ಗೆಟ್ ಟುಗೆದರ್ಸ್ ಅದ ಅಂತ ಹೇಳ್ರಿ ಅಷ್ಟೇ ಅದು ಸಿಂಪಲ್ ಆಗ್ತದೆ. 1 ನಿಮಿಷ ತಡ್ರಿ 1 ನಿಮಿಷ ಗ್ರಹಣದ ದಿವಸ ಟ್ರಾವೆಲ್ ಮಾಡ್ಬೋದಾ? ನಾವ್ ಯಾರು ಪ್ರೆಗ್ನೆಂಟ್ ಇಲ್ಲ, ಯಾರು ಕ್ಯಾರಿಂಗ್ ಇಲ್ಲ.
लिंक खोलो ज्वाइन करो.
ಇಲ್ಲ ಎಲ್ಲರೂ ಔಟ್ ಹೊರಗಡೆನೆ ಇರ್ತಾರ ಅದ್ರ ಟೈಮ್ನಾಗ. ಈಗ ವೆಕೇಶನ್ ಅಲ್ಲ, 2 days ಅದು ಮಧ್ಯಾಹ್ನ ಏನೋ ಸ್ಟಾರ್ಟ್ ಆಗ್ತದೆ ಮಾಡ್ತಿದ್ರು. ಅದು 1 hour 2 hour ಅಷ್ಟೇ ಇರ್ತಾವ. ಅದಾದ್ಮೇಲೆ ರಿಟರ್ನ್ ಮಾಡ್ಬೋದಲ್ಲ, ಬಿಡ್ಬೋದಲ್ಲ ನಾವು ಗ್ರಹಣ ಮುಗಿದ್ಮೇಲೆ. 1 ನಿಮಿಷ ಗೂಗಲ್ ಮಾಡಿ ನೋಡ್ತಿರ್ತಾರೆ ಎಷ್ಟು ಇದರ ಟೈಮಿಂಗ್ಸ್ ಅಕ್ಟೋಬರ್ 25 ಅಕ್ಟೋಬರ್. ಟೈಮಿಂಗ್ ಸ್ಟಾರ್ಟ್ at 5:25 p.m. Solar eclipse will start at 5:25 p.m. on October 24th and it will remain till 4:18 in the evening of ಗ್ರಹಣ ಎಕ್ಲಿಪ್ಸ್, ಸೋಲಾರ್ ಎಕ್ಲಿಪ್ಸ್ ಹಿಂದಿನ ದಿವಸ ಸಾಯಂಕಾಲ ಅಂತ ಕೊಟ್ಟಾರ ಇವ್ರು. ಸೂರ್ಯಗ್ರಹಣ ಹೌದು ವಿಲ್ ಕಮ್ ತಡೆ ಇನ್ನೊಂದು ಬೇರೆ ಟೈಮಿಂಗು ಐತಿದು ಟೈಮಿಂಗ್ ಆಫ್ ಸೂರ್ಯಗ್ರಹಣ ವಿಲ್ ಕಮೆನ್ಸ್ at 12:15 AM. ಓ ಇಂಡಿಯಾ ಅಲ್ಲ ಸಾರಿ 8. Major sites in India. What is the timing of Surya Grahana? ಭಾಳ ನಾನ್ಸೆನ್ಸ್ ಬರ್ತಾವೆ ಜಲ್ದಿ ಜಲ್ದಿ ಬರತ್ತೆ ಹಿಂದಿ ಬರಾಕತ್ತೆ ನೋಡಿ ಹೇಳ 5 minutes.
Morning, all of you. On behalf of the organizing committee, I, Dr. Kamlesh Pallandurkar, welcome you all to the 4-day workshop, Methods in Platelet Signaling and Thrombus Biology of Institute of Medical Sciences, B.H.U. This is the SERB's ICMR-sponsored high-end workshop Karyashala, hosted by Center for Advanced Research on Platelet Signaling and Thrombus Biology at Department of Biochemistry, Institute of Medical Sciences, B.H.U. This will run from 18 to 21st October 2022. We sincerely hope that this workshop will enhance your knowledge and guide you in your future academic research. For the outstation participants, a brief about B.H.U. Banaras Hindu University is one of the most prestigious universities of the country, which was established in the year 1916 by Mahamana Pandit Madan Mohan Malviyaji. It is an autonomous institution of excellence whose visitor is President of India. Dr. S. Radhakrishnan, Dr. C.N.R. Rao, and Dr. Narlikar have been alumnus of this institution. In 1960, the College of Medical Sciences with Professor K. Udupa as its founder principal was established, which further flourished into Institute of Medical Sciences. It is befitting on the occasion that we begin this event by paying homage to Mahamana Pandit Madan Mohan Malviyaji, a great visionary, educationist par excellence, and founder of Banaras Hindu University. Thank you. To grace this auspicious occasion, we have with us our chief patron of this workshop, Professor S.K. Singh sir, Director, I.M.S. B.H.U. and Dean, Faculty of Medicine, I.M.S. B.H.U. Professor N.K. Agrawal sir, Vice Dean, Research, I.M.S. B.H.U. Professor S.P. Mishra sir, Head of Department, and Dr. Josna Kailashia and Dr. Abhijit Mukherjee from Department of Biochemistry, I.M.S. B.H.U. Now, I request Professor S.P. Mishra sir to welcome Professor S.K. Singh sir, Director, I.M.S. B.H.U. with a bouquet. Now, I request Professor Didas sir to felicitate Dr. S.K. Singh sir with a memento. Now I request Professor Abhijit Mukherjee sir to welcome Professor N.K. Agrawal sir with a bouquet. Now I will request Professor Didas sir for his welcome address.
Very good morning and welcome all the participants. And it's a very eventful, auspicious morning for all of us from the Department of Biochemistry that we are welcoming a large gathering online on the subject that is very topical for all of us. It's a methods in platelet signaling and thrombus biology. And on this occasion, we are very honored to have just beside me Professor S.K. Singh, our director, beloved director, and Dean of Faculty of Medicine as well. And beside him, you can see that, can you have the camera here? Just just camera here.
कैमरा दूसरा ही चल रहा है.
From me and from here. So Professor Singh and beside Professor Singh, Professor Neeraj Agrawal, our the in charge of this telemedicine building, and as well as the Vice Dean of Research. So both the people, perhaps you don't know, they are endocrinologist and diabetes specialist. We all have a problem, we run to them, and you know how close diabetes with the platelet and thrombus disorders. They are just hand in gloves to wreck our system. So it is very topical and timely that we have here such an eminent endocrinologist sitting beside ours to inaugurate this occasion. I welcome Sir Professor Singh, as well as Professor Agrawal, and with your grace, let this occasion be inaugurated. Thank you, sir.
Now, I request Professor S.P. Mishra, sir, Head of Biochemistry, to address the gathering.
So, very good morning to all of you. And this workshop is having participants from 200 participants, inclusion of 40 faculty and PhD scholars and postgraduate students. The participation from all parts of the India, pan-India, northeast to Gujarat and from Punjab to Tamil Nadu. This workshop method is platelet signaling and thrombus biology. SERV and ICMR sponsored high-end workshop. Karyasala is a huge learning opportunity for all participants. Though it is an online mode, you will get a total flavor of all techniques being employed in platelet research. I am sure this workshop will pioneer many young researchers to dedicate their life to platelets and thrombosis research. I wish you all success. Thank you.
Thank you, sir. Thank you for your kind words. Now, I'll request our Chief Patron, Professor S.K. Singh, sir, for his words of motivation for the participants.
Thank you. Good morning, everybody. Greetings from Varanasi to all those who are online. My colleagues, Dr. D. Das, Dr. S.P. Mishra, Dr. Pallandurkar, Dr. N.K. Agrawal. I'm sure that this kind of workshop will enrich you for doing good research in platelet signaling and thrombosis or thrombus biology. This very important area, this has a very important clinical relevance, particularly for aging population and population of our country. We know that cardiovascular disease is quite common in India, is more common than the rest of the world because of the transition, because of the rapid growth, industrialization, and the change in the lifestyle. If you have the abnormality in your body with this kind of environment, definitely you will have more problem of cardiovascular disease. And therefore, this is the responsibility of scientists, young generation, to take over research in thrombus biology, platelet signaling, because that can be translated into clinical benefit. And that will help in preventing or treating our patients having cardiovascular disease. So I'm sure this workshop will be a great platform to enrich you in this field and will help you to think, to design research in the area of thrombus biology. And I also hope that this kind of activity will continue in future, that it will not be one time. As we know that science is a dynamic state. A dynamic state, it changes with time. And to update ourselves, this kind of workshop will be required. So I'm sure that you will be having such workshop from time to time. And these are the pioneer in platelet biology will help you to think in that direction. Best of luck. Thank you very much.
Thank you, sir. Your motivational word will definitely encourage our participants, and we will also try to our level best to meet your expectation and do such programs again and again in near future. Now, I'll welcome Professor N.K. Agrawal sir, eminent endocrinologist and Vice Dean Research, I.M.S. B.H.U, to motivate our participants.
Respected Director Sir, respected Dash Sir, Dr. Mishra and Dr. Palandurkar, and all the participants. Greetings from Varanasi again. And this, as we know that Professor D. Dash has been a very ace pioneer in platelet signaling. I have been his student, and so I understand what he has achieved in his lifetime. So it is a commendable for keeping the IMS in the forefront for platelet research. So I think his experience will be benefiting you all, and with his presence, you will get best out of this workshop. So I congratulate Dr. Palandurkar for organizing this, and hope everything is for the best of the science. Thank you.
Thank you, sir.
Now I request Dr. Kamlesh Palandurkar to extend a vote of thanks.
Thank you. Thank you, Dr. Kiran. It is my great privilege to extend the vote of thanks. I would like to thank all the dignitaries, Professor S.K. Singh sir, Dean and Director, Faculty of Medicine, I.M.S. B.H.U, Professor N.K. Agrawal sir, Professor S.P. Mishra sir, Head, and Professor Ragini Srivastava, Dr. Josna Kailashi, and Dr. Abhijit Mukherjee from Department of Biochemistry I.M.S. for their best wishes for successfully conveying this event. I am very thankful to the eminent speakers who have readily agreed to be the part of this scientific grandeur. We have eminent faculties from US and from other parts of the country who will guide our participants from time to time. And I wholeheartedly thank all the participants who had shown their keen interest in the short period of time and shown their overwhelming response for this workshop. Last but not the least, I'll also thank Professor D. Das sir who has initiated planning and he has untiring effort for commencement of this program. I am very thankful to the organizing committee of this event, Dr. Varun Sharma, Dr. Kiran Giri, Dr. Sushil Chaurasia, Mohammad Ikhlaq, and Vipin Singh. In this online event would not have been possible without the Regional Resource Center Telemedicine at I.M.S. B.H.U Varanasi and their in charge Professor N.K. Agrawal sir. I would also try to have a special mention of Rajendraji, Umesh, Akhilesh, and Shashiji from Telemedicine who had helped us to make this program possible. Thank you, sir. Thank you. Thank you. Now, we will move towards the main event of this particular workshop. And in this particular event, Professor Das sir will have his presentation, introduction to the workshop, in which he will tell us about the experimental approach to study the platelet signaling and thrombosis, which are underlying for cardiovascular disorders. Now we'll move towards the main event.
नहीं आप सेट हो रहा है आप बैठिए सेट कर लीजिए ये छोड़ दीजिए थैंक यू वेरी मच थैंक यू सर आई मस्ट थैंक यू सर कुछ बाहर इशारे नहीं हो गया नहीं अभी कितने पार्टिसिपेंट्स हैं 94 अभी जन्म हाँ सर मैं तब तक आपको इंट्रोड्यूस कर दूँ यस सर
म्यूट कर दिया म्यूट कर दिया
जी म्यूट कर दो
Good morning, all of you. Now, the first lecture in this particular workshop will be delivered by Professor D. Das sir. He has been he is an eminent scientist for platelet signaling and biology, recognized in India and worldwide. He is a fellow of National Academy of Science and National Academy of Medical Science. Also, he has many awards and accolades in his pocket. He has been instrumental in converting the lab research into translational one. He has many patents on his name as well. He has been a recipient of many awards of national and international level. He has dedicated his life in platelet signaling and thrombus biology and will definitely guide us through this workshop so that we can, all the participants in the workshop can have the flavor of this particular research. Thank you, sir. Now I will hand over the mic to sir, and sir will guide us in this lecture. Thank you, sir.
Thank you, Kamlesh. Very good morning to all of you. This is the start of this 4-day program, and I am just giving an overview of introduction. And now, though, while you look at the title of the workshop, Methods in Platelet Signaling and Thrombus Biology, you must realize that it is not this workshop is not very much a equipment intensive, equipment specific, because nowadays there are workshops on next generation sequencing, flow cytometry, confocal micro machine equipment, technique specific. This is not that. This is a thematic, a theme based, and the theme is very much outlined, very much. It is based on platelet signaling and thrombus biology. That's it. And if any equipments will come into within this premise, we shall discuss them. As you see the program, there are several equipments, the flow cytometry, confocal microscopy, thromboelastogram, aggregometry, so many things are there. But the basic premise focus remains the same. Now, while having said that, many of you, I think now there are, I see that about 100 or more participants are there on the other side. It will be crossing the minds. Many will be thinking that why I am sitting there in the morning and giving time for 4 days, my precious quality time. Why should I be listening to the thrombus biology platelet? How it will help me? I just looked at, yesterday I had a look at the participants in a very cursory way, and I found that most of them are entry level and biology related. And naturally, I would understand that they would be having to have a career, a research career. That is the goal of any professional in academic life, to have a very successful research goal. So, towards this end, again the question arises, why platelet signaling thrombus biology? I joined in my faculty position, permanent in B.H.U faculty position in 1986. Perhaps many of you are not born at that time. And I also thought at that time, what career I should make. And after a lot of ruling out the things, a lot of considerations, negativity, positivity, I decided to go up for this thing. And many of these considerations stand till today. And that will be the drive perhaps. Many of you may think to have a career in this focused area. Number one, as just in the inauguration, our director, Professor Singh, told that ischemic heart diseases, coronary artery disease, thrombotic, occlusive vascular diseases are the number one killer in our country, in the subcontinent, in globally also. So this being so, if you are starting a research in an area which is highly topical, You can convince the funding agency to give you fund on that reason. You can convince the editors of the journal that you see that I have used this type of drug to ameliorate this type of disease. This disease is so prevalent. So my article may be published. It makes a rational because this is not a very rare disease. Just whatever opposite of the rare, one of the commonest. Anybody after 45, 50, I am now 65. From the, if you cross 45 or 50, you think that मेरा blood glucose बढ़ गया क्या? मेरा BP बढ़ गया क्या? How are my platelets? How healthy, how happy they are? So all our efforts beyond 50, directly or indirectly, they goes towards to keep your platelets happy. You must understand your cells. Their lifestyle, their goal, their aim, what they want, how they want to live, to have a good rapport with them, to have a healthy life. So that makes the cells commonest killer disease in our country. Number one. Number two, why I should do research in this area. Number two, thrombotic disorders, heart diseases, comes under non-communicable, non-infective disorders. Infective disorders now with this corona, etc., they have become little bit in circulation. But always non-communicable diseases are what worry us. Among the non-communicable diseases, what comes? Cancer. Everybody is worried about cancer, whose prevalence is not so much as heart disease. But we worry about cancer. What other psychiatric disorders? What others? Neuro disorders, Alzheimer's, etc., Parkinson's. These are all non-communicable disorders. If you see in our country, how many research institutes are there for these disorders? In cancer itself, in Banaras itself, there are 2 institutes to having hospital treatment as well as research on cancer only. Neurodegenerative disorders. There is one NIMHANS. You can have in Bangalore. You can have a National Brain Research Center in NRC Delhi. Big research institutes to study them. Can you tell me how many research institutes are there country which is dedicated to platelet and thrombosis research? I have been in the area for almost 3 decades. Till now, none. We are doing some scattered work, and some very eminent speakers who will be telling, speaking to you, Professor Guchait from Delhi, Professor Kemparaju from Mysore, and Professor Girish from Karnataka, Tumkur University. Besides 3 or 4 handful of people and Jair Ashraf from Jamia Millia, I don't think anybody will be doing carrying out research. So it comes to your advantage. It is an area where there is a least competition in the country. You write a project, there is no counter project. So that should be another, it is a negative way of thinking, but that should be right. Number 3, why I should do research on this area and waste so much time for 4 days thinking about this. Number 3, just let me have a tea. You want to do research on cancer, start from scratch. You need a culture lab. Or you want to read human tissue, biopsy tissue, to do some histopathology. You want to have a study Parkinson or Alzheimer or something, neuronal cell degeneration type of things. Again, you have to have a culture lab. But for platelet research, what you need? A drop of blood, 5 ml blood, that's it. Either from volunteers or from yourself. Do you need any heavy gadget? No, no gadget for platelet preparation. Do you need a centrifuge machine? Yes, of course. Is it a high-end centrifuge? No. High-speed centrifuge? No. It is a refrigerated centrifuge? A big no. Cold will activate platelets. You should know though, if you want to study a cell, you should acquaint yourself with the cell. Just don't jump into the area without knowing the cell. Platelets get activated in cold. No refrigeration, no refrigerated centrifuge, no high-speed centrifuge. Just a clinical tabletop small centrifuge will be enough to isolate your platelet of the day. Isolate platelet and are some reagents from the market. You purchase adrenaline, put adrenaline, platelet get aggregate. You have controlled platelets, you are aggregated platelets. Sitting on a chamber within 30 minutes, you are having pure platelets, both controlled and adrenaline aggregated platelets. To generate clot, thrombus, do you need any machine? Not at all. You need a small tube. Take the blood, add calcium chloride, drop of calcium chloride. It will clot. No time. Just watch that clot for say 30 minutes. Clot will contract, known as clot retraction. Platelets are highly muscular. They generate a lot of ATP to retract the fibrin strands. You see the drama within your tube. Add your reagents, whatever your area of research, some Ayurvedic drug, this agent, that agent, whatever. Study the effect of clot retraction. This is the beginning. Area, there is no limit. This is the third reason why you can do a research in platelet and thrombus signaling. But there is a caveat. Once I thought all these three reasons in 1986 when I started my research, and I failed miserably. Though I have a very ordinary centrifuge machine, I got blood from my friends, prepare platelets very easily. Platelets are always getting activated. Platelets, you should understand, highly sensitive cells, are sensitive and neuronal cells. It is very easy to activate them, very tough to keep them in a resting state. You should understand this logic. What I understood, because I got all my cells activated at that time. I was very young, assistant professor. There was a conference in Lucknow. On my own money, I attended a conference because one scientist from Netherlands, Professor Joel, was attending that conference. I have read his articles. Nobody is working on platelets in the India. Nobody. So Professor Joel, his literature I knew. I attended a conference in CDRI Lucknow, met Professor Joel, and told that I am starting my career with platelets. I thought it is very easy, but cells are getting activated. So he told me one thing, quote unquote, be gentle with cells. Be gentle with cells. Then he gave me some tips. There are many literatures, etc. You can read how you can be very gentle, very slowly mix the cells. Don't use glass because glass will activate the platelets. Use siliconized glass or plastic wares to prepare platelets. So these are basic things. Once you get good platelets, you can do good research. So these are the 3 points I can tell you why you should do platelet research. And it was, I started with this from 1986. They stand good today also. Now there is a 4th point. And this 4th point is the most important point, why you should utilize your 4 days listening from 10 to 2 p.m. to these lectures. This 4th point is, platelet is a very unusual, queer. Queer means erratic type of cell. They don't follow any rule. No rule of biochemistry they follow. Don't have any nucleus. Lifespan 10 days. If they are circulating, if the aggregate gone. But they are highly sensitive easily activable, as I told you, and there is a signaling hub. Any signaling molecule you pick up, you can try their presence in the platelets. They will interact with many types of molecules. Once, several years back, there is a nano mission. Professor CNRO introduced nano mission in the country, and there is a drive for nano-related research. At that time, I have a student, Sunil. Now he's a faculty in Central University of Punjab. Very good student. And I thought that nano is coming. Try different nanomaterials on platelets. We have not left any nanomaterial. You use nano silver, gold nanoparticles, gold nanorods, nano diamond, graphene, and even this year nano cerium oxide nanoparticle. Whatever you put in the platelets, they will react in some way. You record it and publish it. It will go to a very good journal. Because it is platelets, it is related to thrombus, which is a life and death situation. That is the advantage. You can have a very good identity in scientific circle. Then, several years back, how platelets behave very unusual. Newton has told every bold discovery should have a bold guess. You want to have a very out-of-box discovery. Your thought must be out of box. Don't go by convention. Platelets obey this rogue rule. Platelets are not normal cells, unique cells. Several years back, we have a machine by which we can measure mitochondrial oxygen consumption rate. And one of my students, Vijay, now he's a faculty in B.H.U. He told that, sir, once I stimulate the platelets, they should consume more oxygen because they require more energy. They are activated. But oxygen consumption drops. Unusual. It behaves like Warburg effect of cancer cells. We studied and we published also in 2019 that once you stimulate platelets, they behave like cancer cells. There is no commonness between cancer cells and platelets because cancer cells grow nucleated cells. Platelets have cancer cells almost are immortal. They grow and grow. Platelets have a lifespan of 10 days, no nucleus. This is the only difference between cancer cells and platelets. Rest are same. That is my own belief. Whatever I try, similarity, catch something from cancer cell, try to impose on platelets, they are coming true. That is my secret I can share with you. You can think something in your mind, what happens in the cancer cell, try in the platelet. If you are lucky, you have to have a bold guess to have a bold discovery. If you are lucky, you can find a solution. So, while concluding it, you can look at the this picture here. Is it visible to this slide?
Yes.
Okay, so you can see this picture here, this cartoon. So this is a paper which we published this month only, our latest publication this month in journal known as eLife. Why I am showing this? It is the outcome of this study. What we showed that a protein NOTCH and NOTCH. NOTCH is a protein which is present in stem cells. It is present in cancer. Anti-NOTCH agents are used as anti-cancer drugs. But we just listen to Newton. Just think out of way. We thought that let us try to find NOTCH in platelets. We found it. We did the entire story. It took almost 2 years. Now this story is before you, and this story will, why I projected this story before you, it will show what type of experiments have been involved in this study. Now you can see, this is a platelet, this is a control platelet, so you have to isolate platelets. platelet activation, aggregated platelets. So you have to have an aggregometer or some way to study that platelets are getting aggregated. Now in this aggregation, these are, you see that the fibrinogen is bridging between the surface integrins, how they aggregate. So to show that surface event, something happening, something exposed on the surface, you have to study flow cytometry. Everything will be covered in this workshop. So we did flow cytometry to see that this has happened. Now, this notch protein, these green colors are notch protein. What happens that notch goes to the surface and it has a ligand known as DLL. These blue or parallel ones are ligand DLL. They also go to the surface. So notch makes a link with DLL. So we claim that apart from fibrin bridging, there is another bridging between two platelets, which was yet unknown. This is not DLL bridging. So this is supplementing, strengthening the platelet aggregation. So if you break this bridging by anti-notch agents, it can be anti-platelet. That was the story. It can be anti. So for this, to prove that notch is expressed, you have to do RT-PCR to show the notch transcript are there. We did the RT-PCR. That experiment one has to do. To see the notch expression and DLL4 expression, you have to do Western analysis. We did Western, and we saw that they are abundantly expressed in the platelets. Now on stimulation, they will travel to the surface, translocate to the surface. For that, you have to do flow cytometry. We did and showed that, well, yes, upon stimulation, they are expressed on the surface. Now, again, their presence, you have to do confocal microscopy, something like that, imaging. We did that imaging. To make your story more stronger, authentic, you have to go on adding the experiments and to show that it enhances the clot, you have to do thromboelastogram, many things, etc. And remember, any experiment you do, repeat it as much time as possible to validate your result. Don't jump to the conclusion, be unbiased, be very unbiased. And let me tell you now, the journals, they ask all the raw data. If you are telling that I have did it 5 times Western, you have to give your 5 times raw data to the journals. For example, eLife asked everything from all the raw data. So be very honest, very genuine, never lose your enthusiasm. It will take time, it will try your patience, but if you have a good idea, good initial findings, go ahead. And eventually, you will publish it. Eventually, it will attract fund. Because nowadays, people are only, whenever I visit some institutes, the young generation, we need fund, we need fund. But for that, you have to have a background. Do enough work, have patience, fund will come. But money should not be limited. If you want to collaborate and cooperation, other labs, now we want to have something with the blood bank. Now our blood bank in charge, Professor Sandeep has arrived. So if I want to have a collaboration, go shake hand with another faculty, another laboratory, so long as your story is unfolded. Thank you very much for your patience. And is there another video is there? No.
RS.
So thank you very much. And over to Kamlesh.
Thank you, RS. Somebody is at Chennai, so he will take a lecture from Chennai. Somebody is from Calcutta, so it will continue up to 3 o'clock.
सर ये
All these questions.
नहीं अलाउ कौन करेगा? हेलो हेलो हाँ Okay. Now we invite questions from the audience. Two or three questions will be entertained, and then we will. I will just shortly brief you the guidelines for this particular workshop. Hello. Am I audible? Are there any queries? Please, you are, and also you can write the questions in chat box. मेरा जो ये था
Dr. Sandeep.
कमलेश कमलेश पलंदुकर
मेरा फिगर निकाल दीजिए।
हो जाए।
Am I audible, sir? I have one question.
Please ask your question.
Yes,
So
sir.
what is your,
Sir,
and also
very
mention your
good morning,
name
sir.
and institute. Please mention
Yes,
your
sir.
name and institute.
Yes, sir. I am Shwetanka Prasad and I am from Manipal Tata Medical College, Jamshedpur, sir. Sir, when I will
Yes, ma'am.
isolate the cell from the centrifuge, then after that I have to do the microscopy or I have to do direct flow cytometry, sir.
Direct flow.
What's your good name?
Shwetanka Prasad, sir.
Yeah. So once you isolate your platelets, it depends on which direction you will want to go and whatever facility you are available. I would say that once you isolate platelets, go for cell count. Microscopy cell count, that is how much is your yield, you should know this. That you can do under microscope, you can even counter, you can do a Coulter counter or something, you can count that. Now, if you want to stimulate the platelets, then you can add a stimulant. As I told you, cheapest one is adrenaline, or we use thrombin. If you are from Sigma, etc., you can purchase, or ADP, whatever. If you add that to the platelets, they will activate and aggregate it. Now, once they get activated, activation you can study by aggregometry if you have the machine. If you don't have the machine, no worry. If you have a flow cytometer, then some of the activated platelets express some molecules on the surface. With the flow cytometer, you can study. If you don't have a flow cytometer also, if you have a simple ordinary microscope, activated platelets will project microspines known as philopodia, projections on the surface. They will, like I saw in the cartoon, you can try to look, you should have an eye to look into the microscope and try to find out if how many number of my platelet sample are activated and have projections. So you can have, depending on your availability resource, you can have many things to be done. Many things you can plan out, and you can be innovative also.
Yes,
It is not everything should you innovate yourself. How I can go ahead with this thing. Try to ask questions yourself and try to solve. Okay, thank you.
Yes, sir. Sir, one last question, sir. So these nanoparticles are also activators, sir?
I won't say they are activators. That is a wrong word, because the activators means we till the natural physiological agonists like thrombin, etc. They activate the platelets. But nanoparticles, they interact, and that interaction comes into play because our platelets are all the time there in our blood, and in certain circumstances, nanoparticles can enter our blood. So that is the thing where there will be interface between nanoparticles and platelets. That makes the context of studying effect of nanoparticle on the platelet. That should be contexting. Now, this nanoparticle, once they interact with the platelets, they can be internalized or they can be doing some damage to the cells, and cell will react. All you have to do, you have to document how your platelets behave in absence and presence of nanoparticles, and that documentation is good enough to make a hypothesis.
Thank you so much, sir. Thank you so much.
Welcome.
Sir, good morning. Can I have one question or are we out of time?
There is one question: what is the role of platelet activation in cancer and hypoxia? Okay, let me answer one more question, but there will be specialized speakers for, say, Prasanjit Guchait. He is an expert on hypoxia. So there is a question. Just wait. Let me take this question first. What is how the platelets behave in cancer and hypoxia? You see, once there is a clot is formed, platelets are trapped within the clot, and you see that there is a deoxygenation within the clot. There is a relative hypoxia within the clot. So naturally, the platelets, which are a part of the occlusive, very dangerous type of scenario, those platelets are really hypoxic platelets. So effect of hypoxia on platelets is a very interesting topic. And we have published in 2019, we have shown that hypoxia inducible factor 2 alpha, HIP2 alpha, is expressed on the platelets once they are exposed to platelets. And HIP is bad. HIP is always bad. And regarding the cancer, so these are the things, area can be anything. And regarding the cancers also, platelets are helping in the metastasis. It has already been demonstrated. And and as I told you, platelets resemble many way cancer cells, except that they don't cause cancer. They cause death in another way, more common way. That's the difference.
Are we out of time?
Sir, please, this will be the last one. Then rest of the question, please write in the chat box, and then we'll take all the questions at the end of the day, and or at the end of the session. Okay. So please, and if we are not able to do it in the end of the day, as we know that this is a 4-day workshop, definitely your queries will be solved, and those unanswered at the end of the day will be taken before the start of the next day session. Thank you.
Okay, sir, thank you. Good morning, sir. Thank you for an excellent start talk. I am Dr. Christopher from A.M.S. Kalyani. I am in physiology department. My query is not exactly with respect to thrombus, because I want to keep the platelets, extract the platelets and do some experiment. I don't want to activate them. So my single query is how long we can keep them, and I mean, what is basically the storage modality of platelets without activation? How long we can keep it?
So if you have patients for 1 hour, an expert on platelet storage is sitting just beside Kamlesh. He will explain this way, but I would say that if you isolate platelets, whatever you want to study within 3 to 4 hours. Activation. If you go beyond that at room temperature, they will undergo lesion known as platelet storage lesion. That is, platelet quality deteriorates. That is an area of study itself. You can study that also, because during blood banking in many places, suppose somebody isolated platelet stores in the lab, whatever happens, you can record and report also. That is also area of study. You see what is the background literature, how much it has been done. If nobody has studied that, you can start your study. No worry after all that. But for platelet activity studies, it should be quicker. But how the platelet deteriorate, that part you want to study, keep it for some more time, and you can find the results.
Thank you, sir. Thank you. Thank you.
Thank you, all of you. Now I will just explain the outline of the workshop. So There are some basic rules that we have set up for this particular workshop. The first and foremost rule is there should be no interruption during the lecture. Let the speaker speak his all the mind, and then whatever doubts you have, you can write them down by yourself, or also you can write your doubts or queries in the chat box. All your doubts will be cleared at the end of lecture, taking in sense of the time frame. And if there is a time crunch at the end of the lecture, the doubts and discussion will be cleared at the end of the day. If any query remains unanswered still, so that will be cleared before the starting of the session of the next day. The link for all the days will be the same. It has been sent to all of you. I hope that all of you are able to join. If there is any crunch from your side regarding the network and everything, then please tell us or try to communicate with us on email. A feedback form is a must for that day's attendance. We'll be sharing you the feedback form at the end of the day. Less than 50% attendance will not be provided the certificate, so that we'll know that who are the genuine participants and not. Now, how we are going to move again for this workshop. So as we have completed the first session, that is the inaugural session. Also, we have completed introduction of the introductory lecture by Professor Das sir. Now I'm sharing the outline, and then after that we'll have a small talk on ethical guidelines for the use of human or animal experiment by Dr. Kiran Giri. Then we'll have a guest lecture by Professor Sandeep Kumar sir for platelet and blood banking. Then we'll have a small break of 5 to 10 minutes, and after that we'll move towards the practical session, in which we'll have a small lecture for platelet preparation and application for platelet aggregometer study by Dr. Sushil Chaurasia, and online demo for preparation of platelets, PRP, and platelets from human blood and platelet aggregation will be done by Akla. So, and after that, we'll also have a question answer session for the end of the day. So this is for the first day. Then tomorrow we are going to have, we're starting the session with the guest lecture from Dr. Manas Naik. He's a research assistant of internal medicine in University of Iowa. Then we'll have a guest lecture from Professor Kemparaju sir. He's a professor in Department of Biochemistry, University of Mysore. After that, we'll have our practical session, practical online session that will be delivered by Dr. Josna Kailashia on application of flow cytometry in the study of platelet function and signaling. After that, we'll have an online demo and training. Again, will be conducted by Professor Dr. Josna Kailashia on application of flow cytometry in the study of platelet function and signaling. After that, we'll have the question answer session for the day. On the third day, we have a very packed program. We'll be having the practical session first on this particular day. This will be started with the application of intravital microscopy in the study of platelet thrombus formation in murine model by Dr. Paresh Kulkarni. And after that, we'll have an online demonstration training for in vivo imaging and videography of arterial thrombosis in murine model, that is the mouse model, by intravital microscopy. After that, we'll have two guest lectures. The one by Dr. Prasenjit Gushai from Regional Center for Biotechnology, Faridabad, and then we'll have a lecture and demonstration on thromboelastography by Mohammad Iqla. After that, we'll also have one lecture on application. And on the fourth day, we will have, we're starting the day with session from Dr. Girish, K.S. Girish. He's associate professor in Tumkur University, Karnataka. And we'll have a very good session by Dr. Hina Tabassum, who is a scientist in Division of Basic Medical Sciences, ICMR. And she will guide us regarding how to write and apply for research grant. After that, we'll have a guest lecture by Dr. Shruti Mishra. She is an assistant professor in Bone Marrow and Transplant Research Center, I.M.S. B.H.U. And last, we'll have application of the practical session for application of confocal microscopy in the study of platelet activation, and this will be followed by demonstration and training. Then after that, we'll have a validity session. So thank you all of you. Now I welcome Dr. Kiran Giri for her talk on ethical guidelines.
Thank you, Dr. Kamlesh.
Let me introduce Dr. Kiran. She has been the member secretary of Institutional Ethical Committee, Institute of Medical Sciences, B.H.U for last 4 years. She is also part of Institutional Ethical Committee of Homi Bhabha Cancer Research, Varanasi, and also she has been the gold medalist in MD and has been a very good academician of pharmacology. She will tell us about the use of the ethical guidelines for the use of human blood and also for the animal experiments. Next, please. Yeah. Thank you, Dr. Kiran.
Thank you so much, Dr. Kamlesh, for the kind introduction, and a very good morning to all of you. At the outset, I want to thank the organizer for this opportunity to be part of this scientific endeavor, and myself, Dr. Kiran Giri. The topic given to me is ethical guideline for use of human blood or animal experiments. So the outline of this presentation, as I will confine myself to the blood collection and related ethical guideline, because both animal and human ethical guideline are quite a huge topic. So I will confine myself toward the blood collection, general guideline for blood withdrawn in animal, ethical guideline and amendment in those guidelines. Then we will discuss about the general guideline for blood withdrawn in human and the ethical guideline related to that. So what are the general principles of blood collection in animal? The first and the foremost is that protocol should be approved by the Institutional Animal Ethical Committee. The second one is whenever we are doing the experiment we should plan the experiment in such a way that it should be least painful and stressful, and so that we should add some anesthetic agent like thiopentone, halothane, so that that experiment should be less painful to the animal. And whosoever performing the experiment should have an adequate training for that species, handling of that species. In general, blood sample is withdrawn from the venous, arterial blood and the heart chamber. How much is the frequency? The frequency should be once in 2 weeks in ideal for the non-rodent. All non-terminal blood collection without replacement of fluid is limited up to the 10% of the total circulating blood volume in healthy, normal, adult male on a single occasion, and collection may be repeated after 3 to 4 weeks. In case from the animal we want a sample required for shorter interval of time, multiple sample if we want, then it should be 0.6 ml per kg per day, or 1% of the animal total blood volume can be removed after 24 hours. For example, if I give example of the rat, the blood volume comes out to be 21 ml circulating volume, and the blood volume collection should be up to 1.2 ml, not more than that. And if you are going more than that, then it should be with the fluid replacement. If the study involves repeated blood collection, a sample can be withdrawn through a temporary cannula. The estimated blood volume in adult animal is 55 to 70 ml per kg body weight. Care should be taken for the older and the obese animal. If blood collection volume exceed more than 10% of the total blood volume, fluid replacement may be required, such as lactate ringer solution. Now moving towards the ethical guideline, the first and the foremost guideline which we are following is the CPC SCA guideline. Committee for the purpose of control and supervision of experiment on animal. For toxicology study, this guideline is always very helpful. The guideline from the Organization of Economic Cooperation and Development, they give an extensive guideline for the toxicology study. Now, I will focus on the CPCSE guideline. When we talk about the ethical principle in animal experiment, this comes the very important part, the 4 R's. The first one is replacement. If there is any alternative procedure we can perform, we should go for it. Reduction. Reduction in the number of animal. The guideline says that if we can confine to the 5 animal in a group, or the result can be obtained from the 95 confidence interval, minimum number of animals should be taken. Refinement. The pain should be decreased as much as possible. And last but not the least, a very important one, after the experiment, there should be a rehabilitation for the animal. And CPCA speak much about it, that how the rehabilitation should be done. What are the forms required for the rehabilitation? You must go through all this thing. The CPCA is a committee, and this committee is governed by the guideline that Prevention of the Act, Prevention of Cruelty to Animal Act 1960, and one of the important guidelines, the breeding of an experiment on animal control. These are amendment as a rule in 1998, 2001, and 2006. This is the topic which are covered in the CPCA guideline. Operating procedure, SOPs, different forms are available. The breeding and experiment on rules are available. Form C, Form D, checklist, blood, each and every small form for the weight recording form, blood collection form, hematological parameter form for the animal are detailed and given as an annexure in this guideline. I will focus on the guideline on the regulation of scientific experiment on animal. This guideline extensively tell about the role of institutional animal ethical committee. They say that
ऐसे हम कोई कॉल नहीं करता है.
I request the participant kindly mute yourself. Institutional animal ethical guideline. The member should be duly qualified, the biological scientist, nominee of the CPCSA, veterinarian, the in charge of the animal ethical committee, all this qualified professional to be part of the institutional animal ethical committee. The experiment should be within due care of humanity, under the influence of anesthesia. Experiment of animal are avoided wherever it is possible. There should be the experiment which we are performing should be on the phylogenetically lower animals rather than the larger animals should be avoided. It very specifically tells that not to perform experiment for merely acquiring manual skill, and for that we have the computer assisted learning program in our colleges. And after the experiment, the animal should be properly looked after, and very important that it should be maintained a suitable record of the experiment. Ethical principle adopted by CPCSA for use of animal in scientific experiment. The principle 1 says that the experiment which we are performing should be purpose for the advancement of new discovery for the physiological knowledge, or knowledge which are adding for elevating the surfing of the human being. The principle 2 talks about the lowest on the phylogenetic scale, minimum number, which gives us the valid result at the 95 level of the confidence interval. And if possible, consider for the alternative to the animal experiment. The principle 3 is avoidance or minimization of pain and suffering by using proper anesthetic agent. Aftercare, the principle 4 is about aftercare and rehabilitation, and the principle 5 talks about the duly qualified professional and veterinary care to the animal. The main function of the CPCA is about registration of establishment conducting animal experiment, selection and appointment of the nominee in the institutional animal ethical committee, ethics committee of the registered, regulation of ethics committee of the registered establishment, approval of the animal house facility on the basis of report of inspection conducted by CPCSA, permission for conducting experiment involving use of animal, recommendation for import of animal for use of experiment, and action against establishment in case of violation of any legal norm or stipulation. These are some of the forms which are available, which are related with the blood withdrawn. This form is available in the CPCA guideline. They record the animal number, date, how much quantity of the blood is withdrawn, which anesthetic you have used, for what purpose the blood is withdrawn, and name of the person who have withdrawn this blood with the signature of the animal technician. This record we should maintain for each and every blood collection, for each and every experiment. Similarly, this form is for the hematological record. Following forms are compulsory for the rehabilitation. CPCA guidelines do not end with the experiment only. They also talk about the rehabilitation of the animal after the experiment, and they mention that when the registration during the ethical committee, they mention that which are the NGOs available for the rehabilitation and which are the forms which are required for this rehabilitation is what pre-experimentation done on the animal, experimental detail, post-experimentation detail of animal and suitable certification, and transfer certificate for the rehabilitation. For animal record, this guideline also talks about the when you consider the animal for euthanasia. The animal that is human sacrifice, that is humanly sacrifice of the animal, that the animal is not able to perform its natural function and left in pain and suffering, then you consider it for euthanasia. The animal is exposed to the contagious infectious diseases of zoonotic importance. The animal has been exposed to the radioisotope, then consider an ethical method like cervical dislocation, decapitation, or the carbon monoxide for the euthanasia purpose, suitable method for the euthanasia purpose with minimization of the suffering and pain to the animal. There are certain rules which are amendment considering improvement of the ethical aspect of the research, and the first amendment in this CPCA came as a rule 2, that it should be a significant gain in the well-being of the people of the country. The experiment we are doing should give some significant gain in the well-being of the people of the country. There's certain repetition that insertion of rule line, that minimum number, lower phylogenetic scale. One more important point added is that aftercare and rehabilitation, and not only the aftercare is, they have mentioned it, that the cost of the aftercare should also be included in the project proposal. The cost of the project proposal include the aftercare and rehabilitation cost should be added. It is not. Now, what authority they give to the IAC Institutional Animal Ethical Committee in an amendment brought in 2006, the rule of breeding of experiment on animal, powers to permit experiment small animal were given to the Institutional Animal Ethical Committee. Now, I will not go in detail in all the different aspects of this guideline, but we should be aware of that this guideline also tell about the quarantine period. A very elaborative, extensively given is the good laboratory practices, that how should be the caging of the animal, food supplementation, water supplementation, temperature regulation, humidity regulation, who should be the caretaker of the animal, is given inside the good laboratory practices of the CPCSE guideline. Similarly, this guideline also talks about the biosafety committee of the institute should be established, which take about the genetic research related research proposals, and use of the hazardous chemical related proposal should go under the biosafety committee of the institute. The certain procedures are explained. Terminal blood collection from the dorsal portal vein, retroorbital blood collection. And we owe so much to this small animal, and we should take care, we should ethically perform this experiment. Now, in the next slides, we will move towards the blood collection in human, ethical guidelines related to the blood collection in human. So how much blood we can withdraw for the research purpose? The amounts given is one time 200 cc blood can be collected in a healthy volunteer without further precaution. If we want more than that blood collection, there are certain rules are given that weight should be above 50 kg. The pulse rate must be between 50 to 100 beats per minute. Temperature should not exceed 37 degrees Celsius. The CBC should be withdrawn before sampling. Hematocrit value is given between 0.36 to 0.48. Hemoglobin should be between 12.5 to 20. Total volume from blood must not exceed the 550 cc for one sample. There must be 8 weeks between the sampling. Our next expert will elaborate more on this. Next lecture will elaborate more on this. The blood volume taken from children must be less than 3 cc per kg body weight per 8-week period potential. And if there is potential benefit to the children, 9 cc venous blood can be taken, but with supplementation of the ferrous sulfate, that is 30 milligram per kg per day. Now the ethical guideline, ethical principle of the country which we follow is the ICMR guideline, National Ethical Guideline for Biomedical and Health Research involving human participant. For clinical trial, we follow the guideline, that is the 2019 guideline, New Drug Clinical Trial Act. For more important guideline regarding the ethical practice or ethical conduct of the clinical trial is the Good Clinical Practice Guideline. We'll focus on the ICMR guideline. Not changing guideline. Yeah, my one question to the participant. Many times researcher came to me in the ethical committee and they said that our experiment is nothing but I just want to withdraw some amount of blood from the patient or subject. So what category this ICMR mentioned this, whether it is a less than minimal risk category, minimal risk category, minor increase over minimal risk, or more than minimal risk. Any one of you can answer in chat box. My colleague said it is a minimal risk. Any more answer? Okay, minimal risk. So interestingly, it is minor increase over minimal risk. It does not comes under the category of minimal risk. What ICMR said that when there is only we are recording, probably no harm. For example, if you are collecting a data sample, data available in the public domain, it comes under less than minimal risk. Minimal risk, if you are doing any history taking, physical examination, X-ray, obtaining body fluid without invasive intervention such as hair, saliva and urine sample, it comes under the minimal risk. When we are collecting a blood, it is invasive procedure. It comes under the minor increase over minimal risk or low risk category. So it comes under the category of minor increase over the minimal risk category as per the ICMR guideline. And all other intervention, clinical trials, invasive procedure comes under more than minimal risk or high risk category. So the basic principle I think you all must be aware of, which is the basis of the ethics in our country, is the beneficence. It should be benefited. The research we are doing should be beneficial to the subject. At least we should do no harm. Justice. All have equal opportunity to be part of the research project. Women, some other communities, all should have an equal opportunity to be part of this research. And autonomy, most important one, that they should decide. The ball is in their court that they should, whether they want to be part of this research or not. Autonomy is given to the subject, participant, and these are the basic principle of ethics. There are different types of reviews, for example, full board review, exempted from review. If we are doing a research on the already obtained sample or the sample which are stored in our biobank, and we want to do a research on that, that sample is anonymized sample or leftover sample, then we can ask the ethics committee for an expedited review procedure on that sample. This is given for the BA study, but it is stand to for all the study that blood we are withdrawing should be under the physiological limit irrespective of the study design. The blood we are withdrawing, which we have already discussed in previous slide, in a general guideline that it should be in a physiological limit of the blood collection should be in the physiological limit. Then a recent trend of the stem cell, we all should be aware that what is permissible in our country, that participant stem research is categorized into permissible is adult and cord blood collection, embryonic comes under the category of restricted, and reproductive cloning is prohibited in our country. Areas of research. Biobank are very important. The storage of the blood sample is very important. Biological material, not my field of expertise, but they said that we can store it in the biobank and we can prospectively use that for the research purpose. One important point, whenever we are storing this sample, either we can do it anonymized or unidentified sample already available in the biobank, or we can anonymize that sample reversibly or as well as irreversibly. Coded or reversibly anonymized indirect link of sample is given, which is data to the participant identified with restricted access, or irreversibly identified link to the participant identity is removed and cannot be relinked. Or it can be identify a direct link of sample data to the participant identity exists. So these are the types of sample in the biobank can be stored. Now this is also one of the important aspect we should consider, that is the multi-layered consent. When we are for diagnostic purpose, if we are, or for any research, if we are planning to collect the sample, we can think about this multi-layer consent, which we can apply for the future research area. In this, the participant consent that yes, I am giving consent for this A research, and this can be also used for the future research prospect, or whether they can give consent or they cannot, they can deny the consent. So there is a concept of the multi-layer consent. What is the advantage is that the reconsenting procedure and the recollection of the blood sample can be avoided through this multi-layered consent procedure. Now the last but the utmost important, which I think that we talk about so much about the ethics, but the execution of the ethics comes with the informed consent form. A bilingual informed consent form is the one which execute our principle of ethics and apply it to the research. So what are the important criteria? It is the research statement. It should be clearly mentioned the purpose, duration, procedure. What are the anticipated risks from this research? What are the possible gain from this research? And very important, what are the alternative procedure available that participant can opt for? It should be mentioned in the informed consent form. Confidentiality, it should be mentioned that this is your confidentiality will be maintained. Regarding compensation, the statement should be given in the form. And last, but it is very important that the contact number, not only the name of the person, name, position, contact number of the person should be given in the informed consent form. And lastly, it should be written that the voluntary participant, and at any point of time they can withdraw, they have right to withdraw from this research. One more important thing I want to share with the student, that this informed consent form is not for us only. We should have a copy. The person who is giving the informed consent should have a copy of that in which research they are participating, and whom to contact, and case of any emergency, any adverse effect, whom to contact. So one copy of the form should be given to the participant and one form should be kept with you for the record purpose. And in case of children, if you are withdrawing the blood, assent should be taken. 7 to 17 must assent verbally. 14 to 14 years of age may co-sign the parental consent, and all minor must have a parent or guardian sign a consent form giving permissible to draw blood from their child or for the research purpose. Thank you all. There is a very thin line between ethically doing a research and unethically. What is important is to be aware of the ethical purpose, ethical guidelines which are given, and try to be on an ethical part of the research. Thank you all. I'm happy to have any question related to the confining to the ethical principle.
Phylogenetic relation, animal selection. कुछ नाम वगैरह
Phylogenetic relation. So it means that animal ethical committee is rodent can be comes under the guideline of animal ethical committee. Guinea pig, rats, mice are under the purview of the animal ethical committee. For the higher animals in the phylogenetic relation, we have to take it is not under the guideline of comes under the guideline of animal ethical institutional animal ethical committee. We should take the permission of the CPCA for the higher in the phylogenetic relation.
Madam, good morning.
Very good morning, sir.
I am Dr. Christopher from A.M.S. Kalyani. Could you please elaborate a little bit more on multi-layered consent? You said that one advantage is that recollection of the sample is avoided, which means that we are taking sample for once and we'll be storing it and we'll be using it again. Is that what you meant, or?
No, sir. Yes, sir. You got it right. In a multi-layer consent, you already for one research purpose, for example, I'm doing a cancer research, then I will tell the patient that I am doing a cancer research and I'm taking this for a cancer research purpose, but I will store this sample and I can use for a future research prospect also. So this is the subject may agree to it or may not agree to it, but it is a clearly mentioned consent form which that you will store the sample for the future research prospect also. If you are doing any genetic study, if you want to perform it, that should be very clearly mentioned that for any genetic purpose and clearly not only written in it should be very clearly in their language, should be explained to them that for what purpose it is taken, for what purpose it will be stored. Yes, sir. You got it right, sir.
Yeah. Okay. Thank you, ma'am.
Thank you.
Madam, good morning. I'm Shwetanka Prasad from Manipal Tata Medical College, madam. I have a small query, madam. If I will take my blood, suppose in a EDTA tube, and I will perform it, is it ethical, madam?
Okay. It will come under the purview of your institutional ethical committee. You can do it, definitely. But you will write this in your proposal and you will go to your human ethical committee, and if they allow, you can go for it. They will, for what purpose you are taking, what are the experiment, what are the risk benefit ratio to you, they will check for it and they will give you permission for that.
Yes, ma'am. Thank you, ma'am.
So thank you all.
Hello.
Yes.
Hello, this is Iban Lahadem from A.M.S. Kalyani.
Yes, please.
Yes, actually, my question is, suppose if we have samples collected from patients already, but we have not taken it for research consent, then is it possible that we proceed with those patient sample, or do we need to get consent first from the patient again to proceed with research with those collected samples?
So in ICMR guideline, it is given that if it is an anonymized sample, unidentified sample which is stored in your biobank, then you can ask your ethical committee for waiver of consent. And if they think that they will check for the risk benefit ratio and the purpose of the research, if they think that it is for the research purpose, the research is good for the human being, then they can allow you to use that anonymous sample for your research purpose.
Okay, okay. Thank you.
Waiver of consent in that case.
Okay, thank you.
Thank you so much.
Okay. I would like to mention to all the participants that we will be providing you with the presentation by email. So we will collect all the presentation throughout the workshop and definitely will mail it to you at the end of the day or at the end of the workshop. So don't worry about the materials, and we'll be providing with most of the possible materials for your future references. Now, for the next lecture, I would definitely like to welcome Professor Sandeep Kumar sir, who is Professor and Head, Department of Pathology, Institute of Medical Sciences, B.H.U. He has done his MD from B.H.U. in 2009, and then from 2010, he has joined as a faculty in Institute of Medical Sciences, B.H.U. He had many research publications, almost 50, and also he has organized many conferences of national and international repute. He has also undergone training in many areas related to hematology, and he had membership of Indian Society of Hematology, and he is member of editorial board of Institute of Medical Sciences Journal. He has keen research interest in inherited bleeding disorders and molecular pathology and cytogenetics in hemato-oncology and diagnostics. He had authored some books and chapters as well. I welcome Dr. Sandeep Kumar sir for his lecture on platelet separation and storages for transfusion purposes. Welcome, sir.
Thank you, Dr. Kamlesh. Thanks the organizing committee for providing me or giving me this opportunity. Good morning to all. I'm going to present a talk on platelet separation from the whole blood in the blood transfusion services and its stories. So, blood component separation. So we are collecting blood in blood bag after several meticulous eligibility criteria of the donor. If donor qualify the criteria, then he or she are allowed to donate the blood. And for preparation of different component or separation of different component, there are a large number of instruments are required and a large number of protocols are available for the different component separation for the different purposes. So here we will discuss about the platelet separation and storage. So the blood is collected in a blood bag after fulfilling the eligibility criteria of the donor, and if we need to collect the platelet or separate the platelet from this whole blood, then this blood should be kept at room temperature and it should not be refrigerated in refrigerator. So, and this process should be completed within 4 to 6 hours. And if we do not need to separate the blood, platelets from the blood, then we can keep the blood bag at 4 degrees Celsius and we can proceed for the separation of RBC and plasma. So as per the criteria and need of the society, the national committee says that you have to prepare 100% component from the separated blood so that we can fulfill the need or requirement of the blood and its product for the patient services. So blood component separation, we all know that it is done by the centrifugal force, and all of us know by that we are using some type of centrifuging machine, centrifuge machine, and it is calculated by the radius of that very tube. So here we use two types of spin. One is called light spin or soft spin, and other is heavy spin. Light spin is used for the platelet-rich plasma, for the generation of platelet-rich plasma, and heavy spin for platelet-poor plasma. So here is a sketch that will say when we collect the whole blood, it is kept in anticoagulant and it has some preservative solution. When we do soft spin, then we will divide the blood into two components. One is produced is called PRBC, packed red blood cell, and other is platelet-rich plasma. And for we do hard spin for platelet-rich plasma that will yield platelet-poor plasma and platelet concentrate, and it is kept or it is stored for the supply. And when the platelet-poor plasma is frozen within 6 hours, then it is called fresh frozen plasma, and that can be later on kept at minus 30 degrees Celsius for 1 year. And it can be thawed at the temperature of 1 to 6 degrees Celsius, and we can collect the cryoprecipitate that will use for the bleeding patients and the some liquid portion that will have the cryopoor plasma. This FFP is also, or platelet poor plasma, is also utilized for the generation of some albumin and other proteins by the fractionation methods. When we do hard spin, then it will utilize Buffy-Coat method of preparation of blood component. The Buffy-Coat method of preparation will yield more platelet. So platelet yielded by this method has more number of platelets, and it will after hard spin will yield PRBC, Buffy-Coat, and platelet-poor plasma that is transformed into fresh frozen plasma, and Buffy-Coat is soft spin, and the platelet is collected from that very soft spin Buffy-Coat. So here again the same things, platelet-rich plasma method and Buffy-Coat method, soft spin, and we'll express the PRP, and then the PRP will later on go on hard spin, and will express plasma and separate the platelet concentrate. And in Buffy-Coat method, we later on do the blood bag used in the platelet-rich plasma method and Buffy-Coat method is different. In Buffy-Coat methods, we will use top and bottom blood bag, and in platelet-rich plasma, we use just a normal blood bag. And it has different types of blood bag attached to main or principal or mother bag. So this is this one is this bag. This bag is just collected bag, and this whole blood is centrifuged in a special type of centrifuge that is temperature regulated. And after the centrifugation, we get this type of material. Upper layer is the plasma, bottom layer is the RBC, and this has platelet also. So in Buffy-Coat method, we have a tube, or this is the bottom. So we have a tube connected with this and another bag. So we will collect this RBC from the bottom, and we collect this plasma from the above supernatant, and in between these two we have the Buffy-Coat, and from this we will prepare the platelet concentrate. Here this is the packed RBC, this is the plasma, and this is the platelet. You can see here in the bottom of the platelet bag, this is the platelet material, and it has a plasma volume. So the contamination of the RBC during the separation is minimum. This all the three bags are just separated yesterday. So platelet preparation is done by the RDP method. For the random donor platelet by PRP method, RDP by Buffy-Coat method, another method that is called single donor platelet, and it is separated by the apheresis. So this is the apheresis machine. Here the patient after fulfilling the eligibility criteria allowed to donate the platelet by the apheresis. So it is patient is put on machine, and the whole blood is collected, and from this it is centrifuged inside this, and the platelet is transported here, and the remaining plasma and RBC will be transfused back into the patient, and only platelets are separated. So now the stories, come to the stories. So when we will separate the platelet in a bag, then it should not be stored right just after the separation. It should be kept at the room temperature for 1 or half an hour. Then it will be resuspended in the plasma by gentle mixing. And it is stored at 22 degrees Celsius plus minus 1 degree by constant agitation under the platelet incubator. So agitation during the platelet is important. It helps in the exchange of gases. Otherwise, it will otherwise it is difficult to maintain the pH, and platelet function will deteriorate. So there are different types of platelets, and its quality must be good. So volume by volume, the RDP has mostly 50 to 60 ml of plasma, while by the apheresis or single donor plasma, a single donor platelet apheresis, its volume is between near about 200 to 250. And the volume can be regulated by the apheresis as per the need of the patients. And the platelet count is very good in the apheresis platelet. And it is said that 5 units of or 6 units of RDP is equivalent to 1 unit of apheresis. So pH must be more than 6, and RBC contamination should be very minimal or negligible. And WBC contamination is also very minimal. There is a swirling phenomenon. What is it is when we pass a light beam on the platelet bag, when the platelet is of discoid form, is normal form, resting form, then this causes breaking of the light. And if it is not observed in the blood banking system, that means that unit of the platelet is not good for the transfusion, and its function has lost, and it may be due to the increase of the pH or decrease of the pH. So there are different factors influencing the platelet storage. Declining pH will lead to the change in the shape of the platelets, or you may call it as activation, and loss of the function. So maintenance of pH and function of platelet on the storage depend upon the permeability of the storage bag to oxygen and carbon dioxide, and these platelet bags are porous, and it will help in the permeability of these two gases. So there are different types of literature in the available that will say that we can store the platelet as like the RBC at 4 to 6 degrees Celsius or up to 4 degrees Celsius, and it can be utilized for longer period, as all of us know that the shelf life of the platelet at blood transfusion or 5 days only, but it can be utilized like packed RBC for the 3 months or more. But a lot of work need to be done. But certainly, at storing of the platelet at 4 degrees Celsius is a substitute for the activated platelet when we have a patient, bleeding patient, and that need immediate control of the bleeding, that we can utilize this platelet. But as per the drug and cosmetic rule, it is platelets should be stored at 22 degrees Celsius plus minus 1 degree. So the after storage of the platelet, there are different types of enzymatic activity are going inside the platelet and in the blood bag also. And this says that there is impairment of some aggregation and adhesion function of the platelet. But this is reversed in vivo after transfusion. There are different types of platelet storage lesions. Dr. Das sir has told something during his inaugural speech. So the platelet viability and function decline over time. So, and this is called platelet storage lesion. And there is a good evidence suggesting that many of the platelet storage abnormalities seen in vitro are actually reversible upon the transfusion. There are different types of platelet additive solution. It is a crystalloid and it is used to replace the plasma content of the platelet concentrate. And if we reduce the plasma content of the platelet, that means there is a lot of different types of transfusion reactions can be minimized, because we know that the transfusion reactions are occur due to the antigen or antibody present in the plasma. So there are different benefits, and it lowers the risk of allergic transfusion reactions and equivalent clinical efficacy for the controlling of the bleeding or control of the bleeding. A lower incidence of plasma-associated transfusion reactions like hemolytic transfusion reactions and TRALI, transfusion-associated acute lung injury. And it is also said that the platelet can be stored up to the 12 days. Normally it is stored up to 5 days only. So there are different types of platelet additive solutions are present in the market. So the platelet transfusion is, we know that the platelet doesn't have Rh antibodies, antigen. So the platelet is transfused as per the ABH system or ABO, identical platelet should be transfused to the patient. But in cases of emergency, any group of the platelet can be transfused to any group of the recipient to save the patient. So it is a very scarce human resource. So we should use it very cautiously, as this requires both human and financial investment. And the component used separately or in combination can meet most of the patient transfusion need. And the patient have a very minimum risk of transfusion reactions. Thank you.
I'm very thankful to Dr. Sandeep for his elaborate presentation on blood banking and platelet separation. I now invite all the participants, if they have any query, they can ask Dr. Sandeep for that.
Hello, sir.
Yes.
Please introduce yourself.
Sir, this is Mahesh from University of Hyderabad.
Yes, sir.
Yes. So the very nice presentation, sir. So my question is, so what could be the best methodology to analysis of the metabolites in platelets, sir? Just I want to know this. Metabolomics. Yes, sir.
Metabolic analysis of the platelet.
Yes,
it is we are not performing any metabolic analysis in the blood transfusion services. We are just performing the quality control of that very.
Yes.
But,
this can be answered by the.
May I interrupt on this topic? Yeah. Like if you want to, you want to do activation and then you want to do metabolomics, or you want to just do metabolomics during the particular procedure?
Yeah, we want to see the total metabolic, sir, so during the platelet activation kind of, the phenomenon.
Okay. Then
Yes.
LCMS and NMR both are very good techniques for for these particular type of things. Okay. Then
Okay.
you
Okay.
can you can separate the platelet first. Okay, and then
Okay.
you can go for lysis, and then you can go for analytics. Also, you can do the same procedure after activation and before activation, so that you can actually analyze the different metabolomics which are having the changes during the particular procedure.
Okay. And one more thing, sir, and many times mentioned that. So what is the optimum temperature for research purpose, sir? Just I want to know that.
Yeah.
Optimum temperature to conduct all the research kind of activities. Just I want to.
Optimum temperature.
So that yeah, optimum temperature for storage of these platelets and all. So they are very sensitive. Once you induct for any kind of even for any even light also they can activate and the platelet activation will happen. So I want to know that what is the optimum temperature to conduct all the research activities.
It is room temperature. It is room temperature. When will you have the maximum resting phase of the platelets? When will you reduce the temperature or increase the temperature? Its viability will go.
As sir has told in his slide also that 22 degrees Celsius plus minus 1 degree is the optimum temperature for most of the studies. Yeah.
Okay. And one more question, sir. Just I want to say that RNA signature in the platelets and all. So can you suggest any good method for that?
Yeah. What are you saying?
So just want to see that total RNA transcriptomics. Yeah, transcriptomics.
Yeah.
Yes.
Total RNA. Yes.
Yeah. Yes, sir.
Total RNA.
So I think since it is a anucleated cells, I think it is very difficult to isolate the total RNA. I just want to know what is the best method to RNA isolation and that. So yeah.
Just to in our laboratory, we know that there is a limited transcript in the platelet and genomic DNA, but limited transcript is there.
सिर्फ उसी को करना है तो ये छोटका बनिया का ये वाला जो फोल्डर है
अरे भाई साहब
Sorry, I cannot hear you.
दिमाग लगाते हो
Other participants should mute himself, please.
Yeah.
So Mahesh, just to answer your question that platelets have a limited transcript, there is a limited RNA, but there is a protein synthesis machinery is there. There are ribosomes are there, and very limited translatome is there in the platelets, and these proteins they are synthesized. This RNA is inherited from the parent megakaryocyte. They are carried into the platelets, and platelets synthesize proteins out of this. Now, regarding the isolation of RNA, just briefly, you just one or two sentences. What kit you can use? You can tell that.
Dear sir, for the RNA isolation, we are isolate the was platelet, which counted 4 to 5 into 10 to power 8 per ml. And this 1 ml of this volume I have taken for the RNA preparation by using the trizole method.
Is it clear?
Unmute कीजिए.
Hello sir, is it clear?
Unmute all कीजिए.
Unmute all.
Yeah. Hello.
Mahesh, Dr. Mahesh.
We are audible.
Hello.
कुछ चैट बॉक्स में आंसर दे दीजिए.
सारे लोग म्यूटेड है.
All.
Okay.
अच्छा वो लोग कुछ से म्यूट किया.
Dr. Mahesh, are you able to hear us?
Okay.
Hello.
Protocol दे देंगे.
हाँ. Okay, thank you, sir.
I must thank Professor Sandeep for his elaboratory talk. And now we'll break for 10 minutes. and after that we'll again assemble for the practical session of this for this day. Please rejoin after 10 minutes. Thank you.
नहीं पढ़ रहा है ना?
क्या नाम है?
किस फॉर्मेट में भेजे थे? ये तो बड़ा गैप ज्यादा बन गया है, गैप ज्यादा आ रहा है मेरे में। चलिए मैं करता हूँ चेंज।
कहाँ गए बाहर?
ये
आपका ऐसे
सही करता हूँ।
कॉपी कर लीजिए,
अलग अलग कुछ
12 करिए, टाइम्स न्यू रोमन 12 लोड कर लिए। ये जिटेरो कर दिए है ना ये?
नहीं जुटेरो वैसे नहीं है, वो जुटेरो जब आप खोलेंगे तब आएगा, जुटेरो वैसे नहीं आता।
वर्ड पे जाइए, यहाँ जो दिखा रहा
कहाँ है?
ये जिटेरो करें,
जुटेरो नहीं, डेवलप व्यू, मेलिंग रेफरेंस लेआउट, कुछ गड़बड़ाया है मेरा लग रहा है।
हाँ आपका वो लैपटॉप नहीं सही।
नहीं लैपटॉप नहीं गड़बड़ है।
आप जाइए तो इस पे करिए क्लिक, वही एक मिनट।
जिटेरो
हाँ कहाँ गया जो ABCD लिखा है वहाँ पे?
नहीं ये नहीं आएगा, वो होम पे आएगा ये।
हाँ यहाँ से आपका
1 घंटे में खुल के चला जाए।
ये ऑन हो गया मैं,
ठीक है
स्टार्ट कर।
जी सुशील।
उसी से जाके ये जो लीजरली टाइम है ना, 10 मिनट जो ड्रॉप दे रहे हैं ना,
Yes, sir.
उसमें हम लोग बेटर चैट को आंसर कर सकते।
आप मुझे कौन से पे भेजे थे सुशील?
ये सर आपका ही सर
कौन से वाले 187 पे भेजे थे क्या?
क्वेश्चंस पढ़ सकते हैं, थोड़ा ऊपर स्क्रॉल करो तो फिर
सुशील
आएगा अभी उनका लोग आए।
Hello. I hope most of you are online now. Am I audible? Please write in the chat box. Hello. Yeah. So for the next talk, I'm inviting Dr. Sushil N. Chaurasia. He is scientist C in our Center for Translational Research on Platelet Signaling and Thrombus Biology. He is alumnus of our department only, and his area of interest are platelets biology, thrombosis and hemostasis, and also signal transduction. His current research interests are role of functional notch signaling in platelet activation, development of novel antiplatelet and antithrombotic strategies for management of thrombosis, and role of plasminogen activator 1 in adult and cord blood platelets. He has many publications with high impact journals. He had many awards and also achievements in his pocket, including winner of Young Scientist Award, Best Research Prize winners, and many others. And he is a very dedicated worker of our department. So I request Dr. Sushil to start with this presentation. Yeah, thank you.
slideshow.
Thank you, sir, for giving me this opportunity, and good afternoon to everyone. Thank you, sir, for giving me this opportunity, and good afternoon to everyone. Today I shall talk about how platelet preparation and application of aggregometer in the study of platelet function and signaling. So, so first I will start with platelets. So platelets are small, having 2 to 4 micrometer diameter enucleate cells derived from bone marrow megakaryocytes. Lifespan of platelet is 8 to 10 days, and the essence of platelet function is response to stimuli. So, so once stimulated, platelets rapidly adhere with each other to form microscopic aggregates or the thrombus. So thrombus is a meshwork of polymerized fibrin holding aggregated platelets, which is essential for hemostasis. The main function of the platelet is to stop bleeding by forming aggregates at the site of vascular injury. So, how to prepare platelet from the human blood? So this is the protocol. So first we have to obtain blood samples will be drawn from healthy human participants under informed written consent, and the platelets will be isolated from freshly drawn human blood by differential centrifugation. In brief, peripheral venous blood was collected. Blood will be collected in ACD vial, and then it will be centrifuged at 100 G for 20 minutes to obtain platelet-rich plasma. And this PRP and platelet-rich plasma thus obtained will be centrifuged at 800 G for 7 minutes to sediment platelets after the addition of PG prostaglandin E1 and ethylene diamine tetraacetic acid. मूव नहीं कर रहे. Then then the pellet obtained after centrifugation, then the pellet obtained will be washed with buffer A or wash buffer containing HAPs, sodium chloride, potassium chloride, magnesium chloride, sodium dihydrogen phosphate, and EGTA. And the buffer and the pH of the wash buffer is 6.2, which is buffer A is supplemented with glucose, PSA, and prostaglandin E1. And after and finally platelets will be resuspended in buffer B, which is buffer B or thyroid buffer having composition HAPs of sodium chloride, potassium chloride, magnesium chloride, sodium dihydrogen phosphate, and the pH of buffer thyroid buffer is 7.4, which is physiological pH, which is also supplemented with the glucose. And the final cell count of the platelets will be adjusted in between 2 to 4 into 10 to the power 8 cells per ml by using automated cell counter, and all the steps will be carried out in under sterile condition, and all the precautions will be taken to maintain the cells in the resting condition or resting state. So these are the precautions while isolating platelet. So first one is the avoid shaking of the tubes containing blood sample, because it's shaking mechanical stress causes platelet activation. So pipetting should be done smoothly, very smoothly. Next one is the carefully collect PRP after centrifugation to avoid leukocyte contamination. And cold can also cause activation of platelets. So before isolation, ambient temperature should be maintained. That is 22 to 25 degrees centigrade should be maintained before the isolation of platelet or before collection of the blood. So pH of the buffer should also be maintained, because low pH, because changes in pH can also cause activation or activation of platelet. So the platelets are very sensitive cells. It needs love and affection. If you give love to the platelets, then definitely platelets revert back to you with good results. So whenever you are isolating platelets, be careful about these points. And this protocol is about how to isolate platelet from mouse blood. So first of all, mice will be anesthetized with isoflurane, and then the blood will be drawn drawn through retroorbital vein using heparin coated capillary tubes. So blood will be collected into acetate citrate dextrose vial, citrate dextrose vial diluted with modified thyroid buffer containing sodium chloride, potassium chloride, and sodium dihydrogen phosphate, etc., and also supplemented with glucose and BSA. And the pH of the thyroid buffer is around 7.35. And after blood, then the blood will be centrifuged at 100 G for 15 minutes at room temperature, and the PRP will be isolated. Then add EG1 prostaglandin E1 and centrifuge it at 800 G for 7 minutes to obtain platelet pellet. Now this platelet pellet is again washed with thyroid buffer in the presence of prostaglandin E1 and resuspended in the same thyroid buffer, and resuspend it in the same thyroid buffer. So platelets contribute to hemostasis in two main ways. First one is the formation of primary hemostatic plug, like adhesion, aggregation, and secretion. And second one is the formation of secondary hemostatic plug. In secondary hemostatic plug, there is procoagulant activity are generated, like thromboxane A2 generation, etc. Happens in the secondary hemostatic plug. So platelet granules. Platelet contains three types of granule: alpha granule, dense granule, and lambda granules. So in alpha granules contain fibrinogen, platelet factor 4, factor 5, 1 Willebrand factor, and dense granule contains ATP, ADP, calcium, and serotonin. While lambda granule contains enzymes, it is similar to lysosome and contains several hydrolytic enzymes. So next is the why platelet aggregation testing is needed. So platelet aggregation testing is needed for the evaluation of suspected hereditary disorder or acquired disorder of the platelet function. So hereditary platelet function disorder include defects of addition like Bernard-Soulier syndrome. In this syndrome, there is a lack of glycoprotein 1b receptor, and second one is the defects of aggregation. In case of Glanzmann thrombasthenia, there is a defective integrin receptor, the glycoprotein second B, third A. And more common defects of secretion like alpha granule secretion, dense granule secretion, aspirin-like defects, or other primary secretion defects. Acquired platelet function disorders are more common than the hereditary disorders and include drug-induced platelet dysfunction like NSAIDs, non-steroidal and anti-inflammatory drugs like aspirin and ibuprofen, clopidogrel, various cardiovascular drugs, uremia, and myeloproliferative disorders. So, as I have already discussed, in case of defective addition, in case of 1 Willebrand disease, this is the quantitative as well as qualitative defect in the plasma level of 1 Willebrand factor. But in case of Bernard-Soulier syndrome, there is a lack of the platelet membrane glycoprotein 1b. And defective aggregation like afibrinogemia, deficiency of plasma. In this case, deficiency of plasma fibrinogen is occurs. And in case of Glanzmann thrombasthenia, defective or deficient platelet membrane glycoprotein second and third integrin receptor. So next one is the defective platelet granule secretion. So first one is storage pool deficiency. In this case, deficiency in the dense granule contents like ATP, ADP, and serotonin. Gray platelets. In case of gray platelet syndrome, there was deficiency in alpha granule content like platelet factor 4, platelet 1 Willebrand factor, thrombospondin, and platelet derived growth factor, etc. Arachidonic acid metabolic pathway abnormality can be detected by employing aggregometry, and defective liberation of arachidonic acid from platelet membrane is also studied by using aggregometry. So these are the some examples of selected platelet function defects by employing different types of agonists like ADP, collagen, and ristocetin. So in case of a person having thrombasthenia, when we have performed when we perform experiments with ADP, so we have observed there was a less or decrease in the aggregation was observed. When we have used collagen, there was also a decrease in the aggregation. But when we have performed aggregation with ristocetin, there was a normal aggregation pattern were observed in case of thrombasthenia. In case of thrombocytopathy, when aggregation was induced by ADP, the first phase of the aggregation was normal. When aggregation is induced by collagen, there was a decrease in collagen. There was a decrease in aggregation was observed, and when aggregation is observed by performed by ristocetin, normal aggregation pattern was observed, will be observed. So in case of 1 Willebrand disease, aggregation induced by ADP is normal, by collagen is also normal, and when we have induced aggregation with ristocetin, there was an abnormal pattern of aggregation was observed, will be observed. Non-steroidal anti-inflammatory drugs. In case of person having non-steroidal NSAIDs, anti-inflammatory drugs like aspirin or ibuprofen, the first phase of aggregation when induced by ADP, the first phase of aggregation was normal. When induced by collagen, there was a less aggregation was observed. When induced by ristocetin, then we have observed abnormal aggregation pattern. So principle of the aggregometry. So Principle of aggregometry. Using agonist, we can perform aggregometry by employing agonist or aggregating agents to induce platelet aggregation or cause platelet to reduce their endogenous contents like ADP or ATP. So the platelet aggregation is studied by means of platelet aggregometer. So there are three methods for platelet aggregation. First one is the electrical impedance method. We can perform for this whole blood is needed, and second one is luminescence method. This is also known as lumi aggregometry. In this case, whole blood or PR, we can perform experiments either in whole blood or in PRP or in was platelet. And third one is the optical method. In this case, we have to perform our experiments either in platelet rich plasma or was platelet. So these are the some aggregating agents or agonist. Collagen. The collagen working concentration we can use 1 microgram to 10 microgram per ml for ADP 2 to 10 micromolar, epinephrine 10 micromolar, ristocetin 1.25 mg per ml, arachidonic acid 1 millimolar, and thrombin 0.01 to 1 unit per ml. We can use these aggregating agents to induce platelet aggregation. So first one is the electrical impedance method. The impedance or electrical resistance method of aggregation is non-optical, and in this case, in this method, an electrode assembly is inserted into a cuvette containing a whole blood. The electrode assembly basically consists of two precious metal wires that are immersed in the sample. The instrument measures the electrical resistance or impedance between the two immersed wire. During a brief period of equilibration, a monolayer of platelets are formed on the exposed portion of the wire, so resulting in a stable impedance value. So this stable baseline of impedance is assigned a value of 0 ohms of the resistance. After setting baseline and aggregating agents like ADP or collagen is added to the cuvette, and the platelets are stimulated to form aggregates to the monolayer of the immersed wire. So this accumulation of platelets add electrical resistance to the circuit, which is measured and quantified in ohms. The measurement of the electrical resistance. So the change in the impedance is displayed as a function of the time. So the increase in the impedance is directly proportional to the mass of the platelet aggregates. So if there is more increase in the resistance or impedance, there will also be more platelet aggregates. So these are the benefits of impedance method. So the impedance methods of measuring platelet aggregation allow the study of platelets in more physiologic whole blood environment, while optical method that is not more physiological as compared to impedance method. So impedance aggregation in blood is more sensitive to aggregating effects of ristocetin. So it may be more sensitive to valve infected disease than the bleeding time or ristocetin cofactor assay. Impedance aggregation in blood is not dependent on the optical characteristics of the sample, so test can be performed in lipemic or thrombocytomic samples. And centrifugation is not required in impedance method. So impedance aggregation is especially useful in the condition where mega thrombocytes count is also increased. So this is the picture showing this is the cuvette containing whole blood, and this is the cup and containing this is the cup containing electrodes. This is the this is the membrane, and when we adding agonist of the platelet like collagen and epinephrine, there is you can see here there is a platelet aggregate platelet monolayer are formed and aggregated platelet are seen in this one after adding a collagen or epinephrine or any agonist of the platelet. So this is the data of collagen-induced platelet aggregation in whole blood. So you can, this trace is showing in pre-treated with inhibitor, and this one is vehicle pre-treated. And when pre-treated, when we pre-incubated cells with either platelets, either with inhibitor or vehicle, and then we add collagen, and we have observed this type of pattern. Like in case of inhibitor, there was less aggregation, while in case of control or vehicle treated, there was more aggregation was observed. So next one is the luminescence method or lumiagrigometry. The lumiagrigometer may be used to simultaneously measure platelet aggregation and secretion. The instrument records both aggregation and secretion of the dense granule in this method. The basic principle of the lumiagrigometer is to measure secretion by a sensitive luminescence, that is the luciferin luciferase assay, for the extracellular ATP measurement in combination with simultaneous measurement of the aggregation. The ATP released by platelet dense granule binds with the chronolium, that is the luciferin luciferase reagent, and then generates a light. So this luminescence method, lumiagrigometry is performed by using whole blood or PRP or was platelet. So this modification of aggregation is particularly sensitive to release of granule contents, and is a sensitive measure of the platelet activation. So this is the data showing this data is showing aggregation as well as ATP release. When these two tracing are drug treated and this one is vehicle treated, we have simultaneously measure aggregation as well as secretion, that is luminescence in thrombin induced thrombin induced platelet thrombin induced aggregation. So you can clearly see here when we treated with inhibitor, there was a less aggregation was observed and less ATP secretion was also observed in the same sample. Whereas in case of control sample, there was more aggregation and more ATP secretion was observed. So next one is the optical method. In this method, platelet we can use either platelet-rich plasma or was platelets or was platelets. So platelet-rich plasma, which is turbid in appearance, first of all placed in a cuvette, and the cuvette is warm up to 37 degrees centigrade in the heating block of the instrument, and stirred via a small magnetic bar. Then we have to set baseline light transmittance through the platelet-rich plasma is recorded. The baseline of the light transmittance is set by, in case of when we have performed aggregation with platelet-rich plasma, then we have to set baseline with by employing platelet-poor plasma. So the addition of an aggregating agent or agonist causes the formation of platelet aggregates with corresponding increase in the light transmittance. The change in the light transmittance is converted to the electronic signals and recorded as a tracing by the chart recorder. So this is the data showing trap-induced aggregation of was human platelet by optical method. So these two tracing are the drug treated and this one is the vehicle treated. So you can see here there was more light transmittance, so more aggregation. In case of drug treated, there was less light transmittance and less aggregation was observed. So this is the schematic view. In optical method, there was in aggregometer in an instrument, there was a light source, and this is the cuvette containing platelets in the suspension, and this is the stirrer bar, and this one is the photomultiplier tubes. So you can see here, first of all, when light fall on the, first of all we have to set baseline. So for setting the baseline, we have to use platelet poor plasma. After setting of the baseline, then we have to put platelet poor platelet rich plasma with magnet in the instrument, and then we have to add agonist. You can see here, this is this one shows the platelet aggregation after the addition of the agonist. So first of all, you have to set baseline, then add agonist. So after addition of agonist, there was first phase of the initiation of the aggregation is a shape change. After shape change, there was a primary wave of aggregation, and then after primary wave of aggregation, the release reaction occurs, and then after release of this granule contents, so these granule contents further amplify the aggregation and causes secondary wave of the aggregation. So you can also say this is the resting platelet. This is the resting platelet, and after addition of agonist, there was a shape change and primary wave of aggregation, then secretion, then secreted contents further amplify secondary wave of aggregation. So the 5 possible phases of the platelet aggregation. First, you have to set baseline, then add agonist. After addition of the agonist, there was a shape change, and then primary wave of aggregation was observed. After primary wave of aggregation, there was a secretion of the granule content like ATP and ADP, and these ATP and ADP further amplifies the further amplify the aggregation and causes secondary wave of the aggregation. This is the secondary wave of the aggregation. So platelet-rich plasma aggregometry and the agonist and pattern. So in case of ADP, when we are using ADP as a agonist, there in case of ADP, when you are employing ADP as a agonist and perform aggregometry, you have to observe biphasic curve, that is primary and secondary wave. You can see here is the point of agonist addition, and this is the primary wave of aggregation, and this one is the secondary wave of aggregation. So this shows the biphasic curve. When if ADP is added at too low or too high concentration, it will not get biphasic response. At appropriate concentration, ADP will give biphasic curve. If you are using too low concentration of ADP or too high concentration of ADP, there was a single tracing a single phase is observed. When we are using epinephrine, there was also a biphasic curve was observed. Collagen, there was a lag phase. After lag phase, there was a secondary wave of aggregation second wave is observed. With restostatin, in case of restostatin, there was also a biphasic curve was observed. With thrombin, biphasic curve, and with thrombin, because thrombin is a strong agonist of platelets, so it causes irreversible reaction, irreversible aggregation. And when in case of arachidonic acid, second wave only observed, and in case of serotonin, a primary wave of aggregation with a maximum of 10 to 30% transmittance followed by disaggregation was observed. So how to interpret result? How to interpret data? So platelet aggregation occurs as a two-step process known as primary and secondary wave of aggregation. The primary, in case of primary wave of aggregation, in this case, platelet adhere to another in the presence of external agents like agonist such as ADP, epinephrine, restostatin, or thrombin. And secondary aggregation is characterized as aggregation that occurs after platelets have been stimulated to secrete substances contained in their granules. So it should be noted that some agonists will stimulate primary aggregation and some will stimulate secondary aggregation, while others stimulate both primary and secondary, yielding a biphasic aggregation curve. So in addition, different concentration of the some agonist produce varying patterns of the primary and secondary aggregation. For example, low concentration of ADP induced biphagic, that is both primary and secondary wave of aggregation. Very low concentration of ATP causes induce only primary phase of aggregation followed by disaggregation, and high concentration of ADP जैसे up to 10 microgram per ml induce a single broad wave of aggregation. In case of patient with Glanzmann thrombosthenia, they show in incomplete aggregation with ADP regardless of higher concentration of the ADP. So, in patient with severe von Willebrand disease, aggregation to ristocetin is absent. Decreased of normal aggregation to ristocetin can also be seen in patient with mild von Willebrand disease. Abnormal ristocetin induced platelet aggregation may also be occurs in platelet in patients with Bernard-Soulier syndrome and platelet storage pool defects. So Bernard-Soulier syndrome, platelet aggregating, when you are using platelet aggregating agents, so failure to aggregate in the presence of ristocetin in aggregation by other agonists like ADP. When you are using ADP, collagen or epinephrine, if the person is suffering from Bernard-Soulier syndrome, there is a aggregation pattern by employing ADP, collagen, in epinephrine or normal. Response to low dose thrombin may be delayed. So precaution should be taken while studying platelet aggregation. So aspirin containing compounds should be excluded for at least 10 days prior to the testing, as aspirin interferes with the release reaction. And second was the ingestion of other drugs known to influence platelet function should be avoided for at least the time required for their elimination from the circulation. So these include certain antihistamines, antibiotics, antidepressant, and anti-cancer drugs also. So third one is the lipemia. Lipemia can also interfere with the measurement of the platelet aggregation. So studies should not be carried out shortly after having fatty meal. Many other normal dietary constituents, including alcohol, onions, garlic, peppers, and gingers, may also be inhibit platelet aggregation. So avoid before giving the blood sample. So chilling also activates the platelets. So the blood is processed at ambient room temperature, that is 20 to 25 degrees centigrade. So thank you.
Thank you, Dr. Sushil. You have thank you, Dr. Sushil. You have covered many of aspects of platelet and aggregometry. So I invite all the participants now for their questions, because this is a practical. Previously, we were having the theoretical sessions. So this one is much more practical. After this, we will also show you how we are doing it here in our lab. So definitely all of your questions are most welcome. I think some of the questions are also being taken care in the chat box itself. So definitely, if you have any questions, please feel free to ask.
Sir, Dr. Nisha here on this side. Can you explain once again about the primary and secondary wave that is seen?
Okay.
You are from which institute, ma'am?
Lady Harding, Medical College.
Okay, ma'am. What is your question? Can you please repeat?
Can that first wave and second wave of platelet aggregation be explained once again?
Yeah.
Yes. When when we perform aggregation studies, first of all we have to set baseline. After then we have to add agonist. After addition of the agonist, there was a shape change of the platelets. Platelets changes their shape, and after changing of the shape, then they go for first wave of primary wave of the aggregation. after primary wave of aggregation, there was a secretion of the granule contents, and then secreted contents of the dense granule like ATP and ATP further amplifies the aggregation. That will causes secondary wave of aggregation.
So,
Okay, so thank you.
Okay.
Very good afternoon, sir. Sir, I am
Yes.
Shwetanka Prasad from Manipal Tata Medical College, Jamshedpur, sir. Sir,
Yes, ma'am.
in your this session, sir, this platelet preparation and everything that you have taught us is from the EDTA whole blood, right, sir?
No, citrated citrate blood, acetate citrate dextrose. You can use either acetate citrate dextrose or citrate phosphate dextrose or EDTA.
Yes, sir. So it is in the vacuum tube you have separated, sir.
Yes, I have separated. We have collected blood in acetate citrate dextrose vial.
Sure, sir. Thank you so much, sir.
Yes.
Hello, sir. Hello, sir.
Yes, yes, please.
Yes, this is Mahesh from University of Hyderabad.
Yes, sir.
Just I want to know that this platelet aggregation activity in metabolic disorders will be there, no? So have you done any kind of assess during the patients and all? Patient samples, metabolic disorders like obesity, diabetes, type 2 diabetes, kind of.
No.
Not in patient
Not
sample,
in
but
patient
in vitro. In vitro
Because
experiments.
we have performed all the experiments in vitro conditions.
Okay, okay. And one more thing, you mentioned that. Hello, sir. Can I continue?
Yes.
Please continue.
Yes, and one more thing, you mentioned that plate aggregation test like arachidonic acid and COX axis something.
Yes, arachidonic.
So that arachidonic acid and which COX it is, whether the COX cyclohexane is 1 or 2 kind of any specific kind of thing, or
No specific.
Okay.
There was no specific kind of, but
okay.
Both.
Actually, arachidonic acid is the precursor for many of the prostaglandins and cyclotrins. Okay. So so that is why this COX pathway we need to inhibit, and this is the same mechanism for aspirin as well.
COX.
Yeah.
Oh, yes.
So that is the COX inhibitor, right? Yeah.
Oh, yeah, it's a COX. Yeah. Okay. Thank you, sir.
Sir,
Thank you so much.
Can I have a question, or we are running short of time?
No, sir. Please ask. Go ahead.
Okay, I am Christopher from A.M.S. Kalyani again. My interest is, I am definitely interested in platelets, but not with respect to thrombus or blood clot formation. My area of interest is with respect to mood disorders, with respect to psychiatric patients. So my interest is with respect to serotonin estimation in platelets and serotonin receptors expression with respect to platelets. So my query is, how can we estimate the serotonin levels? Because there aren't much studies from India which basically have estimated serotonin levels in patients and also have a baseline of serotonin levels. Could you please tell me what technique could I use and get these data from, if you can?
You can measure serotonin level by employing KIT, ELISA method. You can
chemiluminescence also works well because of high sensitivity.
Okay.
Yeah. And for that, you can definitely purify the platelet from the sample, and then you use them separately by making lysing.
Lysing. Okay.
Lysing. Yeah.
And last question, this aggregometer, what you mentioned for the with the impedance method, that apparatus, you have customized it or you buy it, or how is that?
We have buy it from Chronolab.
Chronolab
Corporation. Vicon. Yes,
how much estimate?
We will be having the session on the video and session on that is the next session only, sir.
Okay, okay, great. Okay, thank
Yeah,
you.
yeah.
Thank you.
Any more question?
One from the chat. Okay, that I will read. That is from Gaura Hari. That during platelet aggregation study, what matters the most, the quality or quantity? Because if quality is too high with low count, then the clot formed may be small, while the condition with high platelet count and low quality may not stimulate other platelets to form potential clot. Kindly clarify.
You have to make balance in between quality and quantity.
And also, like when you will see in the next video also, that whenever we do experiments, we always do a cell count first, so that we will not be having any wrong notion about the result of the experiment. And always you optimize the count, and then only we proceed for the results. So I think that suffices the clarification.
Sir, I have one question. Myself, Vishnu Priya from IIT B.H.U.
Yes, ma'am.
Sir, we are also doing experiment on platelet aggregation, and we
Okay.
use thrombin for the aggregation process. So,
Okay.
what will be the right ratio of thrombin or the sample of platelets to continue with? Like, means how much? Like you told, 0.01 to 1 unit per ml we can take.
Yes.
And what will be the ratio of platelet concentrate and the thrombin solution?
Concentration of thrombin is totally dependent on the its enzymatic activity. If you are having thrombin, if you are having thrombin, sorry, it depends on the enzymatic activity. Basically, it depends on the enzymatic activity of the thrombin. If it is working well, then you have to use 0.01 unit per ml. It is assay dependent actually.
No, sir. My question is, like I have 1 ml of platelet concentrate, and it is having like 3 into 10 to the power 10 cells.
Yes, yes.
So means how much quantity? Not like I have 0.01 unit per ml solution. So
Okay.
whole 1 ml I have to add, like 10, 20 microliter I have to add, 200 microliter I have to add. Means what will be the quantity ratio?
Quantity, if you are using 1 unit per ml of thrombin, then you have to add 1 microliter of thrombin from the main stock. That stock is 100 unit per. If the stock is 100 unit per ml, then then you have to add 1 microliter per 100 microliter. 1 microliter of thrombin to the 100 microliter of the platelets. So
Okay.
you have to calculate it according to your sample volume
Concentration.
or working concentration.
Okay.
Volume wise, 1 microliter per 100 microliter of your sample works well. For us at
Thank
least.
you, ma'am. Thank you, sir.
Okay. So now whatever are the questions remaining, please write them in the chat box, and we will be proceeding with the next session. That are the virtual lab videos. Okay. So you'll all be able to see what we are actually doing in the lab. Please proceed.
Video होगा video.
Video है.
Drive.
Yeah.
First वाला video.
18.
Dated
नहीं नहीं 18 वाला खोलिए.
Dated 18.
हाँ. So वही 18 वाला पहले.
First 18, the negative.
So this is Mohammad Ikhlaq. He is our main performer of many of the experiments, and yeah.
Pause. Stop हो गया. फिर से restart कर देंगे.
Yeah.
So please
आप भी साइड में बैठो ताकि आप फोकस में निकला नहीं मैम ठीक है क्योंकि
in this video today I am going to discuss about the platelet isolation method from the human blood sample. First of all, I am going to introduce what are the ingredients used in the platelet isolation. First, we draw the blood in ACD well. So we take the 1 ml of the ACD and 9 ml of the blood sample from the human vein, and next we proceed this blood sample to the centrifugation.
प्लीज बीच में म्यूट हो गए प्लीज बोलो
For the platelet preparation, we are collecting the blood from the human sample, and after the collection of blood, then we go for the centrifugation step, and for the centrifugation of the blood, we are setting the centrifuge at 100 acceleration G, 100 centrifugal gravitation. So, and this centrifugation is for 20 minutes at temperature ranging 22 degree to 25 degree centigrade. So, in my lab, we are set at the 22 degree centigrade, and acceleration and deceleration speed. For the acceleration speed, we are using 5, and deceleration we are using 0 for the PRP collection. Then start the next video. Yes. And in this video, you can see there are 2 bottles. There is buffer A and buffer B, as described by the Dr. Susil in previous lecture. So buffer A, in this bottle, we are showing there is a 10x concentration of buffer A and 10x concentration of buffer B. So at a particular bottle, we mentioned here what are the pH of buffer A. Buffer A, we are mentioning 6.2, and for the buffer B, we are mentioning 7.35 to 7.4 pH. So this stock should be diluted at 1x by taking 1 ml of this stock and adding 9 ml of blood, 9 ml of water. And further, this stock should be supplemented with the 5 millimolar of glucose in both buffer A and buffer B. And for the buffer A, we are taking only 0.35% of the BSA, boyne serum albumin. So you can see in this video, there are we are collecting 10 ml of blood. So we are not allow this whole 10 ml of blood to in a single tube. So we separate these blood into 2 Tarsan tube, 15 ml, and which are in balanced way, 5 ml in each tube, and then we allow it to for the centrifugation. So you can see in the corner there is a centrifugation instrument, and this centrifugation we can use this is the cooling centrifugation, but we are not for the platelet preparation, we are not set it at the cooling temperature. We set it at 22 degrees centigrade, and before allowing this sample to the centrifugation, we balance both of these tube in a equal weight by this weighing machine. And you can see here there is the minor disbalance, so we can adjust by the adding of volume from one vial to another vial. Now, after the balancing, we place these tubes into centrifuge. So there are different parameter: speed, time, and temperature, and additional parameters are the pre-cooling and acceleration speed for the speed acceleration and deceleration. So we can accelerate more like 5 to 10 or so. For the platelet preparation, we are using only 5 acceleration in my lab, and for deceleration to collect the PRP, we are using 0 deceleration. So if you are applying the here deceleration like 5 or 2/3, then it will breaks and there is mixing in the blood and the PRP. So that for that reason we are avoiding to mention here any values. So this is the key to set all the parameters. And yes. So this is the temperature parameter. You can see in the 17-degree temperature at this particular time, and this will be risen up for the 22-degree, and time will set at 20 minutes. And there is the RCF value, it is 100. And after checking all the parameter, and we are going to start by clicking the green button. And this complete step, this step complete after 20 minutes. Because we are setting deceleration 0, so it will take about 3 minutes. So after 20 minutes, we have these two oils.
This upper layer is this, lower layer is this. So
you
now
can see,
you can see here, and after the centrifugation, we see the two layer. There is the red one layer. Here, red one layer is the RBC, and buffy coat here is for the WBC, and upper one is the PRP, in which the PRP is the platelet-rich plasma, where the platelets are suspended. So we have to isolate only the PRP. और क्लोजअप एक दिखा दो उसका थोड़ा सा कैमरा को लाके. So in the fresh tube, we collect the PRP. While we are aspirating the PRP, we have to carefully aspirate, because there is we have to avoid the mixing of RBC and PRP. So slowly we are aspirating. So we left till this much volume. We left the now show the PRP. This is the PRP. We are separating from the 5 ml of the blood. Straw yellow. Inverse is present, so the post collecting the PRP, we have to mix it with the EDTA. DTA we are using the working presentation of the 2 millimolar, and PGE1 or we can also use the PGI2, but for the timing we are using the PGE1 1 micromolar in the concentration. So here we take the 2 ml of the PRP, so we have to add the 8 microliter of the DTA and 2 microliter of the PGE1. PGE1 are the inhibitors of platelet secretion, and PGT are the chelating.
PG1 which activate the adenylate cyclase, by which it can increase the concentration of PRP, which further inhibit the release of calcium ion, and that are the regulator, that are the inducer for the platelet activation. So that's why we are using
चल रहा है तो एडिशन ऑफ दिस और लिखा आ जाएगा, एक टेक्स्ट उसको लिख के देना पड़ेगा. The PGE1 valve. So after the mixing DTA and PG1, we are generally mix the PRP, which all the content are mixed homogeneously, and then proceed for the centrifugation. Before centrifugation, first we have to balance the use.
So after PRP collection, now this PRP again we are taking for the centrifugation, and this centrifugation takes at high speed. So we are using in my lab 800 G, and which is for 7 minutes, and temperature is similar as first step.
For the centrifugation to isolate the platelet pellets from the PRP. So this step plasma is getting separated from the platelets. So to separate the pellets of platelet, we centrifuge at the 800 G for the 7 minutes, and the deacceleration is 5.
In this step, we are setting deacceleration as 5 because there is no another cell, only platelets are there. So that's why we are using acceleration 5, and deacceleration is also 5.
This is the second step of the centrifugation, and before pellet is ready, we are going to prepare the buffer. 1x buffer of the buffer A, and 1x combination of the buffer B. Buffer A for the OC buffer, which are using to resuspend the buffer.
ये DTA और PG1 डाल के इंक्यूबेट करते हो?
नो सर,
हाँ,
यूनिट 20.
फिर उसमें क्यों रखा है?
सर दूसरा एक्सपेरिमेंट मेथड से मुझे अलग से एक्सपेरिमेंट करना था, तो रूटीन में वही मेथड चल रहा है. ये पुराना एक्चुअली पेपर का रिवीजन आया हुआ है मेरा, तो उसमें सर ये एक्सपेरिमेंट मेथड को मैं सवाल बहुत पहले सर एक्सपेरिमेंट मेथड प्लेटेड बनता था, तो अभी उसी को मैं बस.
अच्छा, यानी कंपेयर करने के लिए एक्सपेरिमेंट मेथड से और PG1 से.
यस.
ओके, तो दोनों मेथड के में कौन एक्टिवेटेड है या ऐसा है, उसके लिए
कंपेयर करने उसमें कैसा रिजल्ट आ रहा है.
ओके ओके.
Uh, at this time, we are using the PGE1 method to
After the
increase the
filleting,
platelet.
we have to resuspend the fillet of the
At this time, we are using the PGE1 method for the platelet isolation. Uh, before PG1 method, we are using the aspirin method. So what is the difference in between the PG1 and aspirin? So aspirin is block the bind with the cyclooxygenase and covalently by the post-translational modification by the acetylation of cyclooxygenase, and this cyclooxygenase further inhibit the activation of platelets. So, and this is the permanent inhibition of cyclooxygenase. So, however, in case of PG1, and this is not happening. PG1 only activate the adenylase cyclase, and this is not the permanent and covalent interaction of. Uh, this is the irreversible. This is the reversible action. So, which are washed in while we are washing with the buffer A, that is also called wash buffer.
After 7 minutes of the centrifugation, of the PRP, and we have of the buffer A, we are also supplemented with the BSA, 0.35% of the BSA, and in a working concentration, in the working buffer, we are using 5 millimolar of the glucose as for the nutrition supplement for the both buffer A and buffer B. So in the composition wise, buffer A and buffer B both has the same composition, but pH, both have different pH. pH 6.2 to 6.5 pH for the buffer A, and 7.35 to 7.4 pH for the buffer B. After 7 minutes of the centrifugation, of the PRP, and we have of the PRP, and we have a clear observation. We can see here the platelets of the platelet, and upper one is the plasma. So in the next step, after the 7 minutes of the centrifugation, of PRP, we have clear observation. Here is the platelets of the platelet. White is white color in the bottom of the tubes, and supernatant is the plasma. So we have to discard this plasma and resuspend these pellets into the washing buffer called the buffer A, which has a pH 6.2 to 6.5. You can see here we have discarded the plasma, and you can clearly see only the pellets are collected. And now we are going to resuspend this pellet into the buffer A. This is start. So first, before the resuspended in the buffer A, first we have to mix the buffer A supplemented with the PG1 1 micromolar PG1 in whole suspension of the buffer A. And now we add 1 ml of the buffer A. One ml of the buffer A to the platelet pellets. We have to resuspend it very much slowly. I will platelet. So we can see here our pellets are resuspended in the buffer A, which are supplemented with the 0.35% of the BSA and 1 micromolar of the PGE. And now we are going to proceed for the centrifugation. And before centrifugation, we make a bit volume for 5 ml by the buffer A. 1, 2, 3.
So this is the pellet washing steps to remove the extra content of EDTA and PG1, which are EDTA in the which are present in the PRP. So we are adding the buffer A, which is pH 6.2, and this need the resuspension of pellets, and this should be very slow process, which not very fast process. So if we are pipetting very fastly, then platelets getting activated, and that are not used in our experiments.
So after rebalancing again, platelets are being centrifuged.
How many minutes? For this, so briefly we are explaining what are the conditions we are using to separate the platelet from the buffer A. So 800 G for the 800 G for 7 minutes, and temperature is similar, 25 degrees centigrade to 22 to 25 degrees centigrade, and each lesson is 5 minutes. Finding pellets of platelet from the buffer A, and now we have to discard this superintendent of the buffer, and we have the clear pellets which further resuspended into the buffer B, which has the pH 7.35.
So after buffer A centrifugation, we have a pellet of platelet again, which were washed with the buffer A, and now finally I'm going to resuspend this platelet with buffer B, which are also called a thyroid buffer or physiological buffer, which has pH 7.35 to 7.4, and this step is also need a very slow pipetting step. And after the resuspension, now I'm going to count. We are going to count this suspension. What are the concentration of platelets in this content?
We are suspending this pellet into the buffer B, which are a physiological buffer, then the pH 7.34, 35 or 7.4. So we are adding 1 ml of this buffer B, and we have to resuspend it very much slowly. Now you can see here the clear solution of the suspension of the platelet, and there is no any aggregation.
So at the time of resuspension, you can observe by the naked eye there should be an homogeneous suspension, and there is no any particles like aggregates not there in this sample. अभी next video उसी में है, उसको continue. अभी question ले लो. Any questions regarding these videos?
Why pH of buffer A is 6.2, not 7.4?
Huh?
pH of buffer A is 6.2, not 7.4.
So for the pH 6. Acidic pH क्यों है?
Is acidic pH उसका so sorry.
Spontaneous platelet aggregation को prevent करने.
So question is why the pH of buffer A is 6.2, not the 7.4? Because this pH is inhibiting the spontaneous reaction of platelets, so that's why we are not give the physiological situation for the platelet 7.4. So before so to keep them in resting state, we first treat with the buffer having the pH 6.2. Any more question?
Very good afternoon, sir. Shwetanka Prasad from Manipal Tata Medical College once again, sir. Sir, the aspirin will inactivate platelet aggregation, sir, but PGE1 will activate aggregation, right, sir?
No, it's not. both are the inhibitors of platelet aggregation and platelet activation.
In case of when you are using aspirin, aspirin causes irreversible inhibition of the platelet, while when you are using PGE1, PGE1 is reversible.
Yes.
So when you platelet prepared with PGE1 are more responsive to agonist as compared to when prepared with aspirin.
So
Is
both
it
are
clear?
agonists, sir, of platelet.
Both are the inhibitors.
One is cyclooxygenase inhibitor and another one is
Adenylyl
cyclase activator. So PGE1 causes activation of the adenylyl cyclase, and adenylyl cyclase elevates the level of cyclic AMP. Elevated level of cyclic AMP causes inhibited release of calcium, intracellular calcium, and finally leads to the inactivation of the platelets. So this process this is the reversible. While when we are using aspirin, aspirin binds to COX-1 on serine residue, and it will irreversibly inhibit the platelet. So when you are using aspirin, the agonists are less responsive as compared to when you are using PGE1. While performing. So that's why we are avoiding using aspirin.
Thank you so much, sir.
As you know, aspirin is a suicide inhibitor of the enzyme. Okay, so this is the very irreversible type of situation there, and that is why it is not a preferred one for the platelet experiments. Any more questions, please? Okay, so we'll move towards the next. Yeah. Who is this?
Sir, if you want to, sir, myself Vishnu Priya from IIT B.H.U.
Okay, ma'am.
Sir, if we want to activate the platelets, means if we want to proceed with the activated platelets, then there is no requirement to add PGE1, no?
Why you are proceeding with the activated platelets?
Sorry, sir?
In the activated state, PG1 is not very much efficient to work function.
What I think you are asking, when you want to experiment on the activated platelets, why do you want?
Yes, sir.
Why do you have to use PG1? Isn't it?
Yes, sir. Yes.
Yeah. So it's like then you won't be having any baseline data. Okay. If you are starting from the activated platelet itself, then you won't know how much is the activity of the particular agonist or antagonist. You won't be knowing that. Okay. So if you are starting right from the activated platelets, and also don't know the extent of activation. Okay. So that is why we are starting from the baseline. Okay. We are using inactivated platelets, and then we see for the activation. You get it?
But that is for like when we are checking the aggregation, like we have to do some confocal study. Like in that we want to see some interaction with the activated platelets. In that case, we only need the activated platelets. We don't need like that processing and all. So in this case, PGE1 is required.
Listen, there is one more fact. If you have watched the video and closely observed, we are adding PGE1 just before centrifugation. Did you observe? Just before centrifugation. That is the important thing, because we are centrifuging the platelet, it is giving them a stress. If you don't add some platelet inhibitor, then the clump will never be dissociated when you want to resuspend it. Okay. So just before centrifugation, you have to give some inhibitor so that platelets do not clot or do not make aggregate in the platelet pellet. Okay, you have to resuspend the pellet, right? For your investigations, for your experiments. If you don't add inhibitor, you will not be able to dissociate that platelet pellet. You will not be able to make platelet suspension. That is why we are adding inhibitors at every stage of centrifugation, just before the centrifugation. You understand now?
Yes,
Do you
ma'am.
understand?
Thank you, ma'am.
Yeah, once you resuspend the pellet just before experiment, then you don't add anything. other than your agonist or whatever drug or anything you are testing. Then you won't be needing any inhibitor. Okay.
So there is one question in the chat box from Mr. Devesh Kumar, that how can I study platelet dysfunction pathway in a particular disease?
Dysfunction pathway.
So for this dysfunction pathway, like if you are taking the platelet aggregation, so if there is problem in the platelet, there is problem in the person, so platelet aggregation might hyper and hypo condition. Like if you are adding the agonist, the platelet normal aggregation of controlled person, so it is about 80%, and if you are taking the patient which platelet has the less active, so in that particular reason and same concentration of agonist may give the result of only 10% 20%. Sometime if you are in case of low weak agonist, if you are taking the like ADP, so in case of ADP we have two type of pattern in the aggregation graph. First peak and second peak, which are explained by Dr. Susil. But in some patient, so if there is a problem with the aspirin resistance and aspirin sensitive cases, so in case of aspirin resistance person, there is no second wave generated, only first wave is generated. So that we can study by the different way.
Was it related to platelet preparation step? Devesh Kumar asking about platelet dysfunction pathway. Because platelet preparation remains almost same for all samples, other than if you want to do any other experiments. So for aggregation, you can go for whole blood, you can go for washed platelets, or you can go for platelet-rich plasma also. For aggregation, I'm telling you. Any more questions?
I think we can proceed with the next video, and then we can take question after that as well.
Wow. Aggregation. Next video is about the aggregation.
How to perform aggregometry is our next video. Please start the next one.
So here we are showing the count after the preparation of platelet. So we are count the what concentration of platelet present in this suspension. So for the counting we are using this. This is the instrument Coulter principle, and this is from the Beckman Coulter. So you can see there are the two tanks. First one is the electrolyte tank. Here is then waste tank, and here is the aperture through which the cell is passing through. And this aperture works by the single cell. If single cell is passed, then there is the disturbance in the transmission of voltage, and this fluctuation of voltage is recorded by the this instrument, and this further and this final data is recorded in this computer software. So you can see. So for the counting, first we take a 10 ml of the isotonic solution in this vial, and we are adding 10 microliter of platelet suspension, and we mix it 2 to 3 times, and after mixing, we just place it with the aperture, and in the software there is a start key, and we just press it, and there is an open OK option for the sample ID, and after OK we can observe an voltage graph here, histogram, and here and this this picture has then x-axis and y-axis. So in the x-axis there is the concentration, and the y-axis there is the number of cells per ml. So sorry, in the x-axis we are mentioning here the size, and in the y-axis we are mentioning the number per ml of. And for the platelet we are taking concentration of 2 to 4 micrometer of diameter of platelets size, and you can see there is no any other cell contamination here. So generally we are taking 2.5 to 3 into 10 to the power 8 per ml of the cell count to perform our experiment. But for the any special type of experiment we need more or less count. Like if in case of flow cytometer we perform our experiment in 1 into 10 power 7, 6, 7, 8 per ml of the sample. So now sample count is set to the 2.5 into 10 power 8 per ml, and we are going to start our next experiment with the it is aggregometry.
We can measure the percentage of aggregation in wasp platelet as a PRP or in whole blood. And this is the instrument, and it is the chronolog model 700 that we are using for the aggregation. So first we have to demonstrate how can we operate the instrument to for the purpose of aggregation. So there are three different methods we are using for the aggregation. First one is the optical light wave method. Second one is the impedance method, and last one is the luminescence aggregator. So optical aggregator is basically based on the transmission of light, how much percentage of light is getting aggregated, and percent of aggregates are increased, and simultaneously light transmission is increased. While in case of impedance method, we are using whole blood sample, whole human blood sample or whole blood from the mice blood. So in whole blood sample, we are using an electrode. I am showing. So there are two types of electrode. One is a cubit, and it is the single electrodes are there, and another type of is this one. So here is a wire. Sir, here is a wire. That's like zoom video baat. Here is an.
एक सेकंड रुको. हाँ चलो आगे zoom तो थोड़ा सा पीछे हो जाओ. Yes. हाँ बस ओके.
You can see here is a thin.
नीचे. हाँ बस हाँ सही.
There are two thin wires close to each other, which have the clear gap in between these two wires. So once platelets are aggregates, they are fixed in these gaps. So impedance are in this. Here is the development of the resistance, and these resistance are captured in our computer screen. I'll show you later how we are. This is the impedance. In the for the optical method of aggregation, we are using, this is the cuvette, and it is chronolog cuvette, and this is the siliconized cuvette, and which we can process the wasp platelet or PRP, and this cuvette is used to analyze the percent of aggregation optical method or luminescence method for both. So now we are demonstrating the instrument. Here's a passive.
Yes. Zoom करने पे हो जाएगा. ठीक है चलो बताओ. हाँ.
So before we start the aggregation, first we have to set the instrument at the 37 degrees centigrade, and all sample must be at 37. So in the next after the temperature, here is the gain setting. gain setting is basically we use for the luminescence aggregator, not for the optical and not for the impedance method. Gain we can increase here we have set the 0.005 here. We can also increase it till 2. We can increase with the intensity of illuminating reagent. Another one is the speed. While we are incubating the sample, when we have also set the speed of the stirrer bar, which can stir the sample at 1200 rpm. So this rpm is basically for all the method, optical method, impedance method, or luminescence. So for that timing, we are going to proceed a sample for the optical method in PRP for the aggregation. And here is the key. PPP, you have clearly seen here the 1, here right here is 1 and left side is 2. Right side is 2 and left side is 1. And here is come the PPP. This is the reference button. 1 represent, if we set the 1, if we click this button toward the 1, it means ki there are 2 channels in a single machine. One channel we have reference for the both the channel. While we set it is at 2, the reference for each channel individually. Before we start the aggregation, first we have to introduce about the what are the reagents and single machine. One channel.
In this part, we can also see that there are three option more is the mode, set, and select. Select for the this different setting and set for the this particular setting. Mode is to change the optical to impedance. So if you are performing our experiment with the optical Lumi aggregometer or impedance. So for the experiment, then for the different setting, we just click on the mode and we have a different different mechanism for the aggregation.
We set it is at 2 and a star bar. What are the reagents and what are the requirements for the aggregation? So for the optical method, we have to have one cuvette, siliconized cuvette, and a star bar, magnetic star bar, and one is the spacer, another is a pipette, and a reagent that we are using as a agonist. So for the timing we are using the ADP as a agonist in PRP. So here is the PPP and here is the PRP. So for the reference, we are taking 600 microliter of the PPP to the cuvette, and in this cuvette we put it into the reference well. This cuvette is for the reference well. What is PPP? PPP, sir. Now I am taking the PRP.
PPP and PRP, which are PRP is isolated after the first step of the platelet preparation. When we are centrifuging the whole blood, we obtain a 3 layer, PRP layer and buffy coat layer and the whole blood layer. So PRP that are rich in the platelet concentrate. And next is the PPP. PPP is the platelet poor plasma. That plasma which very less in concentration are not presence of platelet concentrate.
And in this cuvette, we take the 250 microliter of the PRP for the aggregation. You can also use the 300 or sometimes use 500 microliter. But 250 are okay for the optical method of aggregation.
So for the PRP aggregation, we are taking 300 microliter or 500 microliter. So for the timing, we are taking the 300 microliter of PRP, and you can see the white color of the stirrer bar. And this is the PPP sample, which is of 600 microliter, placed at the PPP well.
उठाओ उसको हाथ से पूरे इसको हाँ हाँ मोड़ो उसको हाँ आराम से सही है
So here is the two well. One is the PPP well, poor plasma, plated poor plasma well, and here is the PRP well, and we first we put the 2 liter PPP liter, and second the PRP which has the.
अरे हिलाओ नहीं उमेश हाँ
Right. The PRP. This is the setup for now.
सीधा करो नहीं वो बाहर जा रहा है हाँ बस ओके ठीक है हाँ
So this is the software which are used for the data of aggregometer, and we once we open the software, there is the 3 option: run new option is there. So while we are opening this option, so we have an identification part where we mention date or by the name to find out our data after the experiment. And here is the 4 channel: channel 1, channel 2, channel 3, and channel 4. So for the timing, we are performing our experiment in the channel 3. So we just click on the channel 3, and here we mention the sample type and what reagent, what agonist we are using to stimulate the platelet for the purpose of aggregation. So here we are writing here. So this is the PRP. So first we are mentioning here PRP, and because we are using the ADP as a agonist, so we are plus mentioning the ADP. And after setting, we just click on the start, and we can see here there is an scale for the aggregation. So before adding the agonist, we set it to the 0, and we just inform the instrument what is the meaning of 0%, what is the meaning of 100%, and this is done by the reference sample. So after the setting this value, we just stop and click on the rerun, and before clicking on OK, we take the agonist. So we take the agonist regarding our needs. So in these cases, we are using 2 micromolar of ADP concentration. While we are adding the any reagent, we just have any fluctuation in the addition of needles here. You can see here there is the addition and there is the fluctuation of graph, and you can see there is the fast drop in the
yes हाँ बस हाँ सही है.
So you can see in So you can see while we are adding the agonist, you can see there is some it is upright movement of the graph. So this will shows the shape change of the platelets which are present in the suspension. After the shape change, these platelets will aggregate to each other, and while this will aggregating, there is the clear this turbidity of sample is getting down and transmission of light increase. That's why this will down. So we can see here there is a clear 30% of light transmission is there, which shows there is 30% of aggregation of platelets in PRP by this agonist. And again this will reverse, but because we are using the weak agonist, So that why this platelets getting disaggregates again. So this is the that's why this graph is getting up. And you can see here again this will graph is getting again for the in aggregating mode. So this will shows the secondary wave of aggregation, and this secondary wave of aggregation is due to the release of a granular content. There are two type of granules in platelets: alpha granule, dense granule, and these granules releases and which behave like autocrine and paracrine manner and stimulate the further platelets to make an stronger plug up platelets. And this tracing we record minimum for till then 4 to 5 minutes minimum.
सेम चेंज हो सकता है ये जो मे बी अभी मेरे पास एक ग्राफ है यू कैन सी हियर देयर इज
आपका जो अग्निश है वो डिग्रेड हो गया एक तरह से,
इसलिए ऐसा है, इसलिए ऐसा है। अभी मेरे पास एक ग्राफ है वो अभी रिसेंट तक क्या हुआ उसमें से याद है।
so I think we should pause it here.
Yes. So we have find the aggregation graph, and after the addition of agonist ADP.
So this is the another graph which are using with the higher concentration of ADP. So in this graph we are using 5 micromolar concentration of ADP, and we get a stronger induction of aggregation. So first is that there is the shape change, and again first wave of aggregation till there about 45% of aggregation. Again there is the release of the granular content, and there is a lot of granular content released in the surrounding, and this will activate further activation of platelet by the autocrine and paracrine manner, and we have a second wave of graph here.
Second wave of the aggregation.
Okay, sir. Done.
Experimental part to okay.
Any question regarding this aggregation video, please ask.
Unmute.
Unmute all.
Participants can also switch on their videos. So we
Yes.
can also see whom we are talking to.
Yeah. Like Mr. Nilesh has requested for the video. Okay, so we will be providing you with the link where you can because the videos are too big, it will be difficult to provide you all with the videos. Okay, but then we will be providing you with the videos on YouTube. Okay, so this whole session is being recorded and will be available on the YouTube, and then you'll be able to see the videos by yourself. Okay, and at your convenience. I think I guess that will be more useful for you, and you can also do the reruns and whatever you. So that will. And any questions on the aggregometry or again on the platelet count part?
Good afternoon, sir. Once again, Shwetanka Prasad from Manipal Tata Medical College, Jamshedpur, sir. Sir,
yes, ma'am.
At this stage, that Coulter counter, if I don't have it, if we don't have, then we can count it with the cell counter, sir, with microscopy.
Yes, ma'am. The Newberg chamber which is there.
Yes.
I think if it is present with you, definitely you can go for cell count with that. Okay. Previously our lab also did the same, but now we have got the cell counter, so that definitely you can go for Newberg chamber. That will give you precise reading for the cell count. But
Sure, sir. Yes, sir. Sorry, sir.
But then it will be laborious for you. Yes.
Yes, sir. Sir, the second thing, this aggregometry, also some manual method is there, sir, if we are lacking behind the instrument. If
Yes,
we want to do aggregometry.
Yes, manuals are provided by the
Manual
There is
method.
no manual method for
Manual method.
agrigometry. So you have to depend on instrument, and actually
Oh.
instrument is giving you quantitative data, how much
Yes.
percentage aggregation was there. So no other method.
Thank you so much, sir. Thank you so much, ma'am.
Okay, for
Just a sec, ma'am. There are some methods by which you can use the plate spectrophotometer for aggregation studies, but then that will be a qualitative data, not a very quantitative data. And that is, there you can go for the plot formation, everything that you can observe with that. But then that will be a raw data. There you will not get a refined data.
Sure, sir. Thank you, sir. Thank you so much, sir. Thank you.
any more questions, please, related with sample preparation and agrigometry?
Good afternoon, ma'am. It's Gaurav Sahu from Central University of Punjab. So my question is that from the platelet preparation to all through the agrigometry, we have avoiding the use of any silica component like any glass tubes and all those things. But where we are using the agrigometry, we are again using a silica material like the glass cube.
No, it is coated glass. It won't.
Siliconized coated.
Yes, it is siliconized.
Not fully silica made. It is siliconized.
It is specialized product from that instrument company only. So it is compatible with that instrument for aggregation. So you have to purchase that cuvettes also.
It's a part of the consumable.
Those are specialized tubes only for this purpose only.
Good afternoon, sir.
Yes.
Yeah. So have a question. Instead of doing this direct platelets, can I perform all the experiments for flow platelets, where we induce in the cells with PMA treatment and all? No? So can
you please ask again?
Instead of doing direct platelets, all these agrigometry kind of experiments and all, is it possible to perform with pro platelets?
Yes, you can. Yes. You can do that. Pro platelets. The lab synthesized platelets, right?
Yeah, by using that PM induced megakaryocytosis.
Yes, you can do. And actually, this agrigometry is actually used for in vitro generated platelets to assess their function, if they are actually functioning like real platelets. So you can surely do that.
Okay. So and one more thing, is there any difference will come instead of like real pro platelets and completely platelets kind of things? Signature molecules.
In our lab, we haven't done that. So you'll have to depend on other publications for that results.
Okay. Yeah. Thank you so much. Yeah.
Yes, welcome.
Any more questions?
Actually feed delayed है ना, तो कोई पूछता
सर
थोड़ा
टाइप
देर से आता है.
कोई का भी लग रहा है कि कुछ हो रहा है.
We have little bit time, so we can answer some queries. I think official sessions are over, na? So this will be question and answer session.
Yeah. You can ask questions from different section as well if you want to. If you have questions from previous session also, you can ask. No issues.
Good afternoon.
Yes. Please introduce yourself.
Okay, I'm Christopher here from A.M.S. Kalyani. I missed in between because I had to go for lunch, and 2 o'clock I have class. So I just wanted to know one last thing about the aggregometer, the the impedance change, that particular apparatus. How much does it cost? Because that is, I think, if you want to start something with respect to platelets, I think that's the first thing what we would be needing it. Could you please tell me the estimated cost of that?
Sir, we had procured the instrument quite long ago, so definitely we don't know the updated cost, but we'll share with you the contact of the company person so that you can get a quote for yourself.
Sure. Thank you so much. Very informative session. Okay, just because we have class constraints, so that is why we have to teach undergraduates. That is the reason. Thank you so much.
You can email your request to anyone of the organizers so that we remember that we have to send you this information.
Sure. Thank you so much. Bye-bye.
Right now we are having question and answer sessions, right?
Yeah.
So all the answers, all the questions will be entertained now. So you are all free to ask anything about today's all the lectures and videos. If there is no query, we can simply end.
Hello, sir. With continuation of below one, I just want to continue for one more question.
Please.
So as you mentioned that the PRP, the platelet-rich plasma endal, I mean, are you doing any other kind of experiments with PRP kind of thing? I mean, this will translate for another disorders.
Are you asking about disease-specific samples?
Yeah, disease specific. No. If you isolate the PRP endal, plasma-rich plasma endal, so are you, I mean, applying these kind of PRP for other, disorders and like, recently, inflammatory disorders kind of thing? Yeah.
If I'm understanding the question right, you are asking if we can use this PRP samples for other experiments also, other techniques also.
Yeah, not the PRP samples, PRP itself.
PRP itself, right? So yes,
PRP itself.
Experiments and techniques can be used with this PRP also. You can do microscopy, you can do flow cytometry. It depends on your research question, what you want to ask. Like if you want to know,
There are some.
My question is, since the plasma, this platelet-rich plasma is rich in very growth factors, all these kind of things, and no. So, are you proceeding with any kind of, cancer immunoregulation kind of thing, cancer inflammation, like metabolic disorders inflammation? To suppress these kind of things, are you applying, doing any kind of experiment in that?
We have so many other projects. So sample is used for so many projects, and some projects are in the field of cancer biology, newer drug therapies. So we have so many other experiments also, and we use PRP, we use whole blood, we use wash plates. It depends what is the purpose of that particular experiment or research question. Again.
Okay. Why? Because recently I saw that this PRP can use it for hair transplantation and all. So I think probably you might know that.
This is used as a source of growth factor. So yes.
Yeah, growth factors. Yeah, right. Yeah.
Something. Hello, can you hear me?
Yeah.
May I say something about the PRP use? Hello.
Yeah, please.
Can you hear me? In the different departments do the use of the PRP, like dermatology, PMR department, orthopedics.
Yes.
They are doing, and the transfusion medicine provides that the PRP sample after collection from the patient. That is the autologous PRP. So you can consult that any transfusion medicine specialist. I myself worked in the A.I.M.S Jodhpur in the transfusion medicine, and we have done one paper with our faculty regarding the use of the PRP in the derma cases. So that is also published with Saptarshi Mandal sir and from the dermatology faculty Dr. Anil Budania sir. So you can also read that thing. That will be helpful.
Yes.
And we have checked
done
the ELISA for quantitative estimation that was done by me and my junior with my sir, because we have done the ELISA quantitative. So that's why I also interested to attend this session.
Yeah. Sir, very interesting, sir. Nice to talk. Yeah.
I want to ask a question.
Thank you so much.
Yeah, can you hear me?
Yes.
Platelet PRP has been part of sports medicine treatment, rehabilitation treatment for a very long time. So I'm Dr. Hanjubam Barunsam. I am from B.H.U itself, and I see lots of previously before joining here we were part of the different
all the tendinopathies, all the musculoskeletal fasciopathies we treat with PRP because the growth factor of the platelet is important for the regenerative medicine. So not only for PMR, not only for the hair growth, the PRP is basically treatment of all those degenerative tendinopathy part where musculoskeletal pain when injection can be given. And also platelet-rich plasma we can give with leukocyte-rich or leukocyte-poor platelet plasma also there. So depending upon whether you want to include some WBC in the platelet-rich plasma or pure platelet-rich plasma, you can use as a regenerative therapy. So it is a very wide field, it is a growing field, and lots of work has been done. So even if you don't want to see all the platelet detail, the biochemical pathway, you can use as a clinical aspect, and it has been used widely.
Yes, sir.
So this is what I
And
have.
one more thing, it continues for this, sir. And thank you for nice answering and all. And when compared to this and all, compared to the stem cell therapy, so this PRP is more better to the stem cell therapy, or I mean,
Depends upon it. See, for stem cell therapy, it can be mostly stem cell therapy. They are mostly used from, for example, you can take the component from the iliac crest or the bone marrow component. So this all comes under this regenerative medicine, and depending upon your requirement. For example, platelet-rich plasma has been shown to be effective in osteoarthritis case, knee OA. Or if you want to give PRP for like for example for tennis elbow for extensor tendinopathy, that case also there. So but the challenge comes because this all therapy comes under rehabilitation and not yet standardized protocol has not been published. So it depends upon from lab to lab where are used. And regarding which one is better, whether you are going for hyaluronic acid injection or whether you are going for PRP or whether you are going for the bone marrow therapy, it depends upon the clinical findings. So since PRP, the problem with PRP injection is it is very painful in counsel the patient before giving the injection. So since biochemical department has a very rich in platelet, we are planning in the future also in collaboration in working with all PRP therapy, and in the future maybe we will be able to answer your question more appropriately.
Yes, yes, sir. Thank you so much, sir. Thank you, enlighten my knowledge. Yeah.
Sure.
Sir, I have one question. Like we are using PGE1. In place of that, do we have any cost-effective chemical that can suppress the platelet activation? Or can we use the like PGE1 capsules what available in market?
That will be injectable form. That earlier in our lab we also had this cheaper version, as you're saying. We used to have aspirin. So our previous papers from the lab will be describing the platelet preparation methods with aspirin. So you can adapt that process also. It was effective. It was effective for
like if you please you can resort for the PG1 injections. So those will be more effective than the tablet one.
Injectable form.
Yeah.
Aspirin is most cost effective, so and it worked for
What will be the concentration of aspirin we can use, ma'am?
1 millimolar.
Okay, thank you, ma'am.
Will any PRP therapy have in pain management?
Not from our lab. No, we don't do clinical experiments. We are mostly doing either in vitro or cell culture related experiments. We haven't gone for clinical experiments or clinical trials. Not yet.
Okay, thank you.
So regarding this, because I have clinical experience with PRP, so I can answer. In pain medicine, PRP, since I have already told PRP injections are very, very painful. So you have to explain a particular subject if you are injecting PRP, and mostly PRP are injected with, it is not advised to inject PRP with lignocaine or some other painkiller because they will inhibit the platelet and the whole idea will be disrupted. So in pain medicine, as such, PRP is not, it's coming up, it's not widely used, but PRP is mostly used for regenerative musculotendinous condition like osteoarthritis knee, like, tendinopathy, like fascioplantar fasciitis, all these things. For pain management, as such, PRP is not widely used, it is coming up.
Does this answer your question? Yes, so thank you, Dr. Varun, for answering. And to the participants, I want to tell you that he is one of our member of organizing committee, and he will be delivering a lecture tomorrow. So you can have more interactions with him, Dr. Varun, tomorrow also. You can ask him tomorrow. Any other questions?
So if there are no further questions, let me take this opportunity to thank all the speakers, our respected director sir, Das sir, and all the faculty members of the department, and members of the organizing committee for commencement of this workshop. And I thank all the participants for their active participation and patient hearing. We will again start the session tomorrow, right at the right at the time that has been prescribed to you in the event schedule that you have received previously. So thank you all of you, and we will conclude the session for today. Thank you. If you have any queries, feel free to email us on the email that has been provided to you previously. Thank you.
Thank you, sir. Thank you, your team also, sir.
Meeting for all, na?